In the quiet moments before dawn, when the world seems held in perfect balance, countless species are already engaged in an intricate dance of cooperation. While much of ecology focuses on competition and survival, there exists a gentler side to nature, one where organisms find ways to work together, support each other, and create relationships that benefit all involved. Tonight, we'll explore these harmonious interactions, from the smallest bacterial partnerships to the grandest ecosystem collaborations, discovering how life finds ways to thrive, not through conflict, but through connection. In the stillness of dawn, before the first bird calls or the rustle of leaves in morning breezes, an ancient conversation is already underway. It is a dialogue spoken not in words or sounds, but in molecules and chemical whispers that drift through air and soil and water. This is the language of cooperation, the foundation upon which countless partnerships in nature are built. Long before complex animals evolved the ability to see color or hear sound, life was already communicating through chemistry. Single celled organisms developed the ability to detect and respond to specific molecules in their environment, creating the first primitive signaling systems. These molecular conversations allowed early life forms to distinguish between potential partners and threats, to coordinate behavior, and to share resources. Today, billions of years later, these same chemical principles underlie every cooperative relationship we observe in the natural world. The air around us, seemingly empty and silent, carries thousands of these invisible messages. A meadow in bloom releases volatile organic compounds that travel for miles on air currents, each molecule precisely crafted to attract specific partners. The sweet fragrance of jasmine at twilight is not merely pleasant to our senses, it is a chemical beacon calling to night flying moths, promising nectar in exchange for pollination services. The earthy smell after rain carries chemical signals from soil bacteria and fungi, announcing their presence to plant roots seeking symbiotic partners. This chemical communication system operates with remarkable specificity, like keys designed for particular locks. Signaling molecules bind only to matching receptors on their intended recipients. A plant producing compounds to attract nitrogen-fixing bacteria will synthesize different molecules than one seeking mycorrhizal fungi. The precision is extraordinary, with organisms able to fine-tune their chemical output based on their current needs, the season, and even the time of day. Consider the underground world, where plant roots engage in constant chemical dialogue with the soil community. Root cells secrete flavonoids, strigolactones, and other signaling compounds that serve as recruitment advertisements. These molecules announce the plant's willingness to trade sugars and other resources for specific services. Bacteria that can fix atmospheric nitrogen respond to one set of signals, while fungi capable of extending the plant's nutrient gathering network respond to another. The root system becomes a chemical bulletin board, posting specific job openings for different types of microbial partners. The evolution of these signaling systems represents millions of years of refinement. Random chemical production would be wastefully expensive, so natural selection has favored organisms that can communicate efficiently and accurately. Plants that produce the right signals to attract beneficial partners while deterring harmful ones gain significant advantages over their less communicative neighbors. Over time, these advantages accumulated, leading to increasingly sophisticated chemical vocabularies. Visual signals add another rich dimension to cooperative communication. The bright colors and intricate patterns of flowers serve as advertisements visible from great distances, But these displays are far more complex than simple attraction mechanisms. Petals often contain ultraviolet patterns invisible to human eyes that create landing strips and directional guides for insect visitors. The size and shape of flowers provide information about the type and amount of reward waiting inside. Trumpet shaped blooms suggest deep nectar reserves accessible to long tongued pollinators, while flat, open flowers offer easily accessible rewards for short tongued visitors. The timing of these visual displays reveals sophisticated coordination between partners. Morning glories open their petals precisely at dawn, coinciding with the activity patterns of their bee pollinators. Evening primroses reserve their display for dusk, when sphinx moths begin their nocturnal flights. This temporal synchronization minimizes wasted effort and maximizes the likelihood of successful encounters between partners. Behavioral signals create yet another layer of cooperative communication. The famous waggle dance of honeybees demonstrates how animals share detailed information to benefit their partners. When a scout bee discovers a rich nectar source, she returns to the hive and performs a ritualized dance that encodes both direction and distance to the flowers. The angle of her dance relative to vertical indicates direction relative to the sun, while the duration of her waggle run communicates distance. Other bees decode this information and fly directly to the resource, benefiting themselves, their colony, and the plants they will pollinate. These communication systems often create networks that extend far beyond simple paired relationships. When one species signals the discovery of resources or the presence of threats, others may eavesdrop on these conversations and adjust their behavior accordingly. A bird's alarm call warning of a nearby predator may cause multiple species to take cover, creating temporary multi species alliances for mutual protection. Chemical signals released by damaged plants can alert neighboring plants to increase their defensive compounds, creating waves of preparedness that ripple through plant communities. The biochemical pathways underlying cooperative signaling are marvels of molecular engineering. Plants don't simply dump random chemicals into their environment. Instead, they synthesize specific signaling molecules through carefully regulated enzymatic processes. Each step in these pathways is controlled by genes that have been shaped by natural selection to optimize cooperative outcomes. The precision extends to timing and concentration, with organisms able to adjust their chemical output, like adjusting the volume on a radio, to ensure their messages reach their intended recipients without attracting unwanted attention. Trust presents a fundamental challenge in any cooperative relationship. How can organisms ensure their partners won't cheat or exploit their generosity? Evolution has crafted several elegant solutions to this dilemma. Many cooperative relationships involve ongoing exchanges where both partners continuously provide benefits. If one partner begins to cheat, the other can immediately respond by reducing their contribution, making dishonesty costly and ultimately self-defeating. Some relationships have evolved elaborate verification systems. Legume plants that partner with nitrogen-fixing bacteria don't simply take their partner's word for it. They monitor the actual nitrogen production in their root nodules and adjust their resource allocation accordingly. Nodules that produce more nitrogen receive more resources from the plant, while less productive nodules are abandoned. This creates strong incentives for bacterial partners to maintain their side of the bargain. The sophistication of cooperative communication is perhaps best illustrated in relationships that have been co-evolving for tens of millions of years. Certain orchids have evolved flowers that precisely mimic female wasps, complete with visual appearance, chemical scent, and even tactile texture. Male wasps, deceived into attempting to mate with the flower, inadvertently collect pollen and transport it to other flowers. While this might appear deceptive, it actually represents highly refined communication where the plant provides exactly the sensory stimulus needed to trigger beneficial behavior in its partner. Modern research techniques are revealing new layers of complexity in these ancient communication systems. Chemical analysis can now identify and quantify the specific molecules involved in interspecies signaling. Genetic studies trace the evolutionary history of signaling pathways, showing how they've been modified and refined over time. High-speed photography captures behavioral interactions in unprecedented detail, revealing subtle cues and responses that were previously invisible. The flexibility of these communication systems is particularly striking. Many organisms can adjust their signaling based on past experiences, current conditions, and the availability of alternative partners. A plant experiencing drought stress might increase the sugar concentration in its nectar to make itself more attractive to pollinators, compensating for reduced flower production. A well-nourished plant might reduce these costly investments, conserving resources for other needs. This behavioral flexibility allows cooperative relationships to adapt to changing environmental conditions and maintain their effectiveness across varying circumstances. As darkness falls across the landscape, cooperative communication continues in new forms. Night blooming flowers release intensified fragrances into the cooling air, calling to nocturnal pollinators through chemical signals that travel farther in stable nighttime air. Bioluminescent bacteria coordinate their light production through chemical signaling, creating synchronized displays that serve various cooperative functions. Root systems increase their chemical activity in the absence of photosynthesis, coordinating with soil organisms to optimize nighttime resource sharing. The language of cooperation is both ancient and eternal, a molecular conversation that began billions of years ago and continues today in every ecosystem on Earth. In soil and air, in fresh water and ocean depths, organisms are constantly communicating their willingness to cooperate, their ability to provide services, and their need for partnership. This invisible network of chemical, visual, and behavioral signals forms the foundation upon which nature's most remarkable partnerships are built. Understanding this language opens our eyes to the cooperative nature of life itself. Where we once saw only competition and conflict, we now recognize sophisticated systems of mutual aid and shared benefit. The morning fragrance of flowers, the complex patterns of migration, the intricate relationships between predators and prey, all reveal themselves as components of a vast cooperative enterprise, where success comes not from defeating others, but from finding ways to work together. In this gentle chemistry of cooperation, we find the true foundation of ecological harmony, the molecular basis for the partnerships that sustain life on our planet. In the warm, crystal clear waters above a coral reef, a remarkable scene unfolds with the precision of a well choreographed ballet. A large grouper, easily the length of a human arm, hovers motionless near a coral outcropping, its mouth agape and gill covers flared wide. Tiny fish, no bigger than a human thumb, dart fearlessly in and out of those powerful jaws, picking away parasites and dead tissue with delicate precision. This is nature's cleaning service in action, a partnership so essential that entire sections of coral reefs are dedicated to facilitating these life-sustaining encounters. The relationship between cleaner fish and their clients represents one of the most visible and well studied examples of cooperative service relationships in nature. These cleaning stations, scattered throughout coral reef systems worldwide, operate like underwater health clinics where large fish, rays, and even sea turtles queue patiently for their turn to receive grooming services. The small cleaner fish, primarily wrasses and gobies, have evolved specialized feeding behaviors and anatomical features that make them perfect partners in this arrangement. The blue-streaked cleaner wrasse, perhaps the most famous of these service providers, advertises its profession through distinctive coloration and behavior. Its bright blue and black stripes serve as a uniform, instantly recognizable to potential clients throughout the reef. When a large fish approaches a cleaning station, the cleaner wrasse performs a distinctive dance, swimming in small loops and displaying its bright colors to signal its availability and peaceful intentions. This behavioral advertisement is crucial as it allows the cleaner to distinguish itself from similar sized fish that might be seen as prey. The trust required for these relationships to function is extraordinary. Large predatory fish must suppress their natural hunting instincts to allow tiny fish to swim directly into their mouths and gill chambers. The cleaners, meanwhile, must navigate the dangerous confines of their clients' bodies, working in spaces where a single reflexive bite could end their lives. This mutual trust has been built over millions of years of coevolution, resulting in behavioral modifications on both sides that make these partnerships possible. Client fish exhibit remarkable behavioral changes when visiting cleaning stations. Normally, secretive species emerge from hiding places and hover in open water, making themselves conspicuous and vulnerable to predators. Aggressive territorial fish temporarily abandon their hostility, allowing cleaners to work without interference. Some clients even change color, darkening or lightening their skin to make parasites more visible to their cleaners. These behavioral modifications represent significant investments in the cleaning relationship, demonstrating just how valuable these services are to reef fish communities. The cleaning process itself is methodical and thorough. Cleaner fish systematically inspect their clients from head to tail, removing external parasites, dead scales, and infected tissue. They pay particular attention to areas their clients cannot reach themselves, such as the inside of the mouth, the gill chambers, and wounds or abrasions on the body surface. Some cleaners specialize in particular types of clients or specific types of cleaning tasks, creating a diverse service economy within the reef ecosystem. Beyond the obvious health benefits, these cleaning relationships provide additional advantages to both partners. For the cleaners, client fish represent a reliable and renewable food source. Rather than hunting for scattered prey items, they can establish territories around cleaning stations and wait for food to come to them. The high-protein diet of parasites and dead tissue supports rapid growth and reproduction in cleaner fish populations. For the clients, regular cleaning reduces parasite loads, prevents infections, and may even improve hydrodynamic efficiency by removing drag-inducing growths and damaged scales. The importance of cleaning relationships extends far beyond individual health benefits. Research has shown that removal of cleaner fish from reef sections leads to significant changes in fish community structure. Client fish begin to abandon areas lacking cleaning services, leading to reduced biodiversity and altered ecological dynamics. This demonstrates that cleaning relationships are not merely convenient arrangements, but essential components of reef ecosystem function. Similar cleaning relationships have evolved independently in ecosystems around the world, revealing the fundamental importance of these services across different environments. In the kelp forests of the Pacific coast, senorita fish provide cleaning services to larger fish species, operating cleaning stations among the swaying kelp fronds. In freshwater environments, various species of small fish and even some invertebrates have evolved cleaning behaviors, serving as mobile hygiene services for larger aquatic animals. The terrestrial world offers equally fascinating examples of cleaning partnerships. The relationship between oxpeckers and large African mammals represents one of the most visible cleaning services on land. These small birds spend their lives riding on the backs of buffalo, rhinos, giraffes, and other large herbivores, feeding on ticks, flies, and other parasites that plague their hosts. The birds gain a steady food source while providing their hosts with relief from parasitic burdens that can cause anemia, disease transmission, and constant irritation. Oxpeckers have evolved specialized adaptations for their cleaning lifestyle. Their sharp claws allow them to grip firmly to their host's hide, even when the animals move through thick brush or break into sudden runs. Their pointed beaks are perfectly shaped for plucking parasites from hair and skin crevices. Perhaps most remarkably, they have developed the ability to walk and even sleep while clinging to moving animals, treating their hosts as mobile territories rather than temporary perches. The relationship between oxpeckers and their hosts involves complex behavioral negotiations. Large mammals must learn to tolerate the bird's movements across sensitive areas of their bodies, while the birds must be careful not to irritate their hosts by being too aggressive in their feeding. Some hosts actively solicit cleaning services by positioning themselves in ways that give the birds access to heavily parasitized areas. Others use subtle body language to indicate when they've had enough grooming for one session. Recent research has revealed that oxpeckers provide services beyond simple parasite removal. They often serve as early warning systems, alerting their hosts to approaching predators or other threats through distinctive alarm calls. This additional benefit transforms the relationship from a simple cleaning service into a more complex partnership involving both healthcare and security services. In the Amazon rainforest, army ants maintain cleaning relationships with various species of birds that follow their raiding columns. These ant-following birds, including antbirds and wood creepers, don't actually eat the ants themselves, but instead capture the insects, spiders, and small vertebrates that flee from the approaching ant swarms. In return, the birds inadvertently provide cleaning services to the ant colonies by removing parasites and debris that might otherwise accumulate in the ants' temporary camps. The world of invertebrate cleaning services reveals partnerships of astounding delicacy and precision. Cleaner shrimp in tropical marine environments provide grooming services to fish, removing parasites with their specialized feeding appendages. These tiny crustaceans establish cleaning stations in caves and crevices, advertising their services through distinctive waving motions of their long white antennae. Fish queue up at these stations just as they do at cleaner fish territories, demonstrating that the fundamental principles of cleaning relationships transcend taxonomic boundaries. Some cleaner shrimp have formed partnerships with sea anemones, establishing their cleaning stations within the protective embrace of the anemone's stinging tentacles. This arrangement provides the shrimp with protection from predators while they work, allowing them to focus entirely on their cleaning duties. The anemone benefits from the increased fish traffic as some potential clients occasionally become meals when they venture too close to the anemone's tentacles. The evolution of cleaning relationships involves intricate co-evolutionary processes where both partners continuously refine their roles and behaviors. Cleaners evolve improved abilities to locate and remove parasites, while clients evolve better ways to communicate their needs and tolerate the cleaning process. This ongoing evolutionary dance results in increasingly sophisticated partnerships that benefit entire ecological communities. Seasonal variations add another layer of complexity to cleaning relationships. In many ecosystems, parasite loads fluctuate throughout the year, creating periods of high demand for cleaning services, alternating with times when such services are less crucial. Some cleaning partnerships are maintained year-round, while others are seasonal arrangements that coincide with peak parasite activity or breeding seasons when animals are particularly vulnerable to infections. The study of cleaning relationships has revealed important insights into the evolution of cooperation and trust in animal societies. These partnerships demonstrate that even between species with very different evolutionary histories and ecological roles, mutual benefit can overcome potential conflict and create stable, long-lasting relationships. The success of cleaning services in diverse ecosystems suggests that cooperation may be more fundamental to ecological organization than previously recognized. Climate change and human activities pose new challenges to cleaning relationships. Ocean acidification and warming temperatures stress coral reef ecosystems, potentially disrupting the delicate balance of cleaning stations. Habitat fragmentation on land can separate cleaning partners or eliminate the specific environmental conditions that make cleaning services possible. Understanding and protecting these relationships becomes increasingly important as we recognize their essential role in maintaining ecosystem health and biodiversity. As moonlight filters down through the water column above a coral reef, the day shift of cleaner fish gives way to nocturnal cleaning services provided by different species. Cleaner shrimp emerge from their daytime hiding places, setting up nighttime cleaning stations where different fish species come for services under cover of darkness. This continuous cycle of cleaning and grooming ensures that the health maintenance needs of reef communities are met around the clock, a testament to the fundamental importance of these cooperative relationships in maintaining the complex web of life that thrives in our planet's most diverse ecosystems. The gentle efficiency of nature's cleaning services reminds us that cooperation and mutual care are not human inventions, but fundamental principles woven into the fabric of life itself. In the soft light of early morning, a single apple blossom trembles gently in the breeze, its 5 white petals forming a perfect landing platform around a center rich with golden pollen and sweet nectar. This simple flower represents one of nature's most essential partnerships, a relationship so fundamental that without it, our world would be barren of fruit, vegetables, and countless other plant species that depend on animal partners for reproduction. The art of pollination is perhaps the most visible and celebrated of nature's cooperative relationships. Every flower that blooms, every bee that buzzes from blossom to blossom, every butterfly that sips nectar from a meadow wildflower participates in an ancient dance of mutual benefit that has been refined over more than 100 million years of coevolution. This partnership between plants and their animal pollinators has shaped the appearance of our world, creating the spectacular diversity of flowering plants that paint our landscapes in ever-changing palettes of color and form. Plants face a unique challenge in reproduction. Unlike animals that can move to find mates, Plants are rooted in place, unable to travel to deliver their genetic material to potential partners. The evolution of flowers represents nature's ingenious solution to this problem of immobility. Rather than moving to their mates, plants learn to enlist mobile partners to carry their pollen from flower to flower, transforming the challenge of plant reproduction into an opportunity for mutualistic cooperation. The basic architecture of this partnership is elegantly simple. Plants invest energy in producing attractive flowers, nutritious nectar, and protein-rich pollen. Animals visit flowers to harvest these rewards, inadvertently collecting pollen grains on their bodies and transporting them to other flowers where fertilization can occur. Both partners benefit. Plants achieve reproduction while animals obtain high-quality food resources that fuel their own survival and reproduction. But within this simple framework lies extraordinary complexity. The diversity of pollination relationships reflects the remarkable creativity of evolution in solving the basic challenge of moving pollen from one flower to another. Different plants have evolved flowers tailored to attract specific types of pollinators, while pollinators have developed corresponding adaptations that make them more efficient at harvesting floral rewards. Consider the intricate relationship between orchids and their pollinators. These plants have evolved some of the most sophisticated flowers in the plant kingdom, with intricate shapes, patterns, and scents designed to attract specific pollinators. The bucket orchid of Central America creates a temporary trap for its bee pollinators, forcing them to climb through a narrow passage that ensures they contact both the pollen-bearing anthers and the pollen-receiving stigma. The bee receives a dose of fragrant compounds that it uses to attract mates, while the orchid achieves precise pollen transfer. Deep tubular flowers like those of honeysuckle and trumpet vine have co-evolved with long-tongued pollinators such as hummingbirds and sphinx moths. The flower's shape ensures that only animals with the appropriate feeding apparatus can access the nectar reward, while the positioning of reproductive parts guarantees that successful feeders will contact pollen. This mutual specialization creates highly efficient pollination systems where both partners have evolved to maximize their benefits from the relationship. The timing of flowering represents another dimension of pollination partnerships. Many plants coordinate their blooming with the life cycles and activity patterns of their pollinators. Spring wildflowers burst into bloom just as hibernating bees emerge and begin foraging for the protein-rich pollen needed to start new colonies. Evening blooming plants like 4 o'clocks and evening primroses time their opening to coincide with the activity of night-flying moths, their primary pollinators. The chemistry of pollination reveals layers of sophistication that extend far beyond simple nectar production. Many flowers produce complex bouquets of volatile compounds that serve as long-distance attractants, allowing pollinators to locate flowers from considerable distances. These scent signatures are often highly specific, helping to ensure that pollinators visit multiple flowers of the same species rather than wasting pollen by transferring it to incompatible plants. Some plants have evolved to manipulate their pollinators' behavior through sophisticated chemical deception. Certain orchids produce compounds that mimic the sex pheromones of female wasps or bees, causing males to attempt copulation with the flowers. During these futile mating attempts, the male insects become covered with pollen, which they then transfer to other flowers as they continue their search for real mates. The nutritional quality of floral rewards plays a crucial role in maintaining pollination partnerships. Nectar is not simply sugar water, but a carefully balanced solution containing sugars, amino acids, vitamins, and other nutrients that meet the specific dietary needs of target pollinators. Hummingbird pollinated flowers typically produce nectar with sugar concentrations around 25%, ideal for the high energy demands of these tiny birds. Bee pollinated flowers often include amino acids in their nectar that complement the proteins bees obtain from pollen. Pollen itself serves dual roles in pollination relationships. For plants, it carries genetic material essential for reproduction. For pollinators, especially bees, pollen provides high quality protein necessary for larval development and colony growth. Some plants produce two types of pollen: fertile pollen for reproduction and sterile feeding pollen, rich in proteins and fats, that serves purely as a nutritional reward for pollinators. The morphology of pollinators reflects their coevolutionary history with flowers. The long proboscis of a sphinx moth perfectly matches the depth of the flowers it visits. The branched hairs on a bee's body create an electrostatic charge that helps attract pollen grains. The specialized bill shapes of different hummingbird species correspond to the flower types they prefer, from the straight bills of bee hummingbirds that feed at short, straight flowers to the curved bills of hermit hummingbirds, adapted for accessing curved or hanging blossoms. Pollinator behavior demonstrates remarkable learning abilities and memory that enhance the efficiency of pollination partnerships. Bees can learn to associate specific flower colors, shapes, and scents with high quality rewards, becoming increasingly efficient at locating and accessing their preferred flowers. This flower constancy benefits both partners. Bees maximize their foraging efficiency, while plants increase the likelihood that their pollen will reach compatible recipients rather than being wasted on flowers of different species. The social organization of some pollinators adds another dimension to plant pollinator relationships. Honeybee colonies communicate information about flower locations through the famous waggle dance, allowing successful foragers to recruit nestmates to productive flower patches. This recruitment system can rapidly direct hundreds or thousands of bees to flowering plants, providing plants with intensive pollination services while giving the bee colony access to abundant resources. Seasonal changes create dynamic patterns in pollination networks. Spring blooms provide early season resources for emerging pollinators, while late season flowers sustain pollinators through autumn and provide resources for overwintering or migration. Plants that bloom at different times throughout the growing season create temporal niches that support diverse pollinator communities and reduce competition for pollination services. Migration adds a continental dimension to some pollination relationships. Monarch butterflies, ruby-throated hummingbirds, and other long-distance migrants create moving networks of pollination services, connecting plant populations across vast geographic ranges. Plants along migration routes provide essential refueling stops for travelers while receiving pollination services from visitors that connect them to distant plant populations, promoting genetic exchange across landscapes. The vulnerability of pollination relationships to environmental change has become increasingly apparent. Climate change disrupts the careful timing that coordinates flower blooming with pollinator activity. Habitat fragmentation separates plants from their pollinators, breaking links in pollination networks. Pesticide use can harm pollinators directly or make flowers toxic to visiting insects. These disruptions threaten not only individual species, but entire ecological communities that depend on pollination services. Conservation efforts increasingly focus on maintaining pollination networks rather than protecting individual species in isolation Creating pollinator corridors that connect fragmented habitats allows both plants and pollinators to maintain their partnerships across human modified landscapes. Reducing pesticide use in agricultural areas protects the insects that pollinate both crops and wild plants. Urban gardens and green spaces designed with native plants provide stepping stones that support pollinator populations in developed areas. The economic value of pollination services has brought new attention to these relationships. Crops worth billions of dollars annually depend on animal pollinators, making the conservation of pollination partnerships an economic as well as ecological necessity. The decline of managed honeybee populations has highlighted our dependence on these relationships and spurred research into alternative pollinators and pollination management strategies. As evening settles across a flowering meadow, day shift pollinators give way to their nocturnal counterparts. Hawk moths unfurl their long tongues to probe deep into moonflower blossoms that opened with the coming darkness. Bats visit agave flowers that bloom in desert nights. These nighttime partnerships continue the ancient rhythm of pollination that has sustained flowering plant diversity for millions of years. The art of pollination reminds us that beauty and function are inseparably linked in nature. The flowers that delight our senses exist not for our pleasure, but as advertisements and rewards in ongoing negotiations between plants and their animal partners. In every blooming garden, every flowering tree, every wildflower meadow, we witness the continuing success of one of evolution's most creative and mutually beneficial partnerships. The gentle hum of bees among flowers, the flash of a hummingbird's wings at a trumpet vine, the delicate dance of a butterfly from bloom to bloom, all speak to the enduring power of cooperation in the natural world. In these partnerships, we find not competition but collaboration, not conflict but mutual care, revealing that the foundation of life's abundance lies in the capacity of different species to work together for their common benefit. High in the canopy of an Amazonian fig tree, a toucan plucks a ripe purple fruit from among the broad leaves. The sweet flesh provides the bird with essential sugars and nutrients, but hidden within that succulent reward lie dozens of tiny seeds, each one carrying the genetic blueprint for a new fig tree. As the toucan flies through the forest canopy to its next feeding site, these seeds travel safely within the bird's digestive system, beginning a journey that may carry them miles from their parent tree to locations where they can establish new populations. The partnership between plants and the animals that disperse their seeds represents one of nature's most elegant solutions to the challenge of reproduction and survival. While pollination enables plants to create seeds, seed dispersal ensures that offspring can establish themselves in new locations, reducing competition with parent plants and colonizing suitable habitats that may be far from their origins. This relationship has shaped the evolution of fruits, the behavior of countless animal species, and the very structure of ecosystems around the world. The development of fleshy fruits represents one of the plant kingdom's most successful investments in animal partnership. Rather than simply releasing seeds to fall randomly around the parent plant, flowering plants learn to package their seeds within nutritious, attractive rewards that would entice animals to carry them away. The energy cost of producing these fruits is substantial, often requiring plants to divert significant resources from growth and other activities, but the benefits of effective seed dispersal far outweigh these costs. The diversity of fruit types reflects the remarkable variety of dispersal partnerships that have evolved between plants and animals. Small, brightly colored berries, like those of elderberry and serviceberry, are perfectly sized for songbirds, with colors that stand out against green foliage to attract avian partners. The birds consume the entire fruit, seeds and all, gaining valuable nutrition while providing the seeds with safe passage through their digestive systems to distant locations. Larger fruits serve different partners and achieve different dispersal outcomes. The massive fruits of trees like Brazil nut and cocoa require larger animals capable of handling their size and weight. Monkeys, large birds, and terrestrial mammals become the primary dispersers of these seeds, often carrying them to different forest layers or habitat types, where the seedlings may face different competitive pressures and environmental conditions. The timing of fruit ripening reveals sophisticated coordination between plants and their animal dispersers. Many plants ripen their fruits to coincide with periods when their target dispersers are most active and in greatest need of nutrition. Autumn fruiting plants in temperate regions provide critical resources for birds preparing for winter migration, while the animals provide seed dispersal services just before the onset of conditions that could help seeds overwinter and germinate in spring. Some plants have evolved to produce fruits in sequential waves, extending the period of interaction with animal dispersers and increasing the likelihood that seeds will be transported to various locations under different environmental conditions. This temporal spreading of fruit production also helps ensure that not all seeds are consumed during a single feeding event, protecting against the risk of total reproductive failure. The biochemistry of fruit ripening demonstrates the sophisticated communication that underlies seed dispersal partnerships. Unripe fruits are typically green, hard, and often bitter or toxic, deterring consumption before seeds are ready for dispersal. As seeds mature, fruits undergo dramatic changes in color, texture, aroma, and taste that signal their readiness to animal partners. The bright reds, purples, and yellows of ripe fruits serve as visual advertisements visible from considerable distances, while the development of sweet sugars and aromatic compounds provides additional attractants and nutritional rewards. The journey of seeds through animal digestive systems involves remarkable adaptations on both sides of the partnership. Seeds must survive the mechanical grinding, acidic conditions, and enzymatic breakdown that characterize animal digestion, while plants must balance seed protection with the need to make fruits digestible and attractive. Many seeds have evolved protective coats that resist digestive processes, allowing them to pass through animals unharmed while the surrounding fruit flesh is digested and absorbed. Some seeds actually benefit from their passage through animal digestive systems. The scarification that occurs during digestion can weaken seed coats in ways that promote germination, while the brief exposure to acidic conditions may trigger biochemical processes that prepare seeds for sprouting. The deposition of seeds in nutrient-rich animal waste provides an additional advantage, giving germinating seedlings access to nitrogen and phosphorus that may be limiting in many soils. The spatial patterns of seed dispersal create genetic connections between plant populations that might otherwise remain isolated. A single frugivorous bird may visit multiple plant populations during its daily foraging, mixing seeds from different sources and facilitating gene flow across landscapes. This genetic mixing helps maintain the health and adaptability of plant populations while enabling species to track environmental changes by establishing new populations in suitable locations. Migration adds a continental dimension to seed dispersal relationships. Birds that travel thousands of miles during seasonal migrations, carry seeds across vast geographic ranges, connecting plant populations across continents and enabling rapid colonization of distant suitable habitats. The timing of fruit ripening often coincides with migration periods, providing migrating birds with essential fuel while giving plants access to long distance dispersal services. Different animal dispersers create distinct patterns of seed distribution that influence forest structure and plant community composition. Birds tend to deposit seeds in open areas or forest edges where they frequently perch and defecate, creating clusters of bird dispersed plants in these transitional habitats. Mammals may carry seeds deeper into forest interiors or to specific microhabitats like den sites or regular travel routes, resulting in different spatial distributions of mammal dispersed species. The size and behavior of disperser animals influence both the distance seeds travel and the habitats where they are deposited. Small songbirds may disperse seeds only a few hundred meters from parent plants, but they visit a wide variety of microhabitats and can access locations unavailable to larger animals. Large mammals like elephants and tapirs can carry seeds many kilometers from their sources and often deposit them in clearings or disturbed areas where establishment success may be particularly high. Some plants have evolved sophisticated strategies to manipulate the behavior of their dispersers, maximizing the effectiveness of seed dispersal. The fruits of many rainforest trees ripen asynchronously, with only a few fruits becoming available at any given time. This forces frugivorous animals to visit multiple trees and travel greater distances between feeding sites, resulting in more extensive seed dispersal than would occur if all fruits ripened simultaneously. The nutritional composition of fruits reflects the specific needs and preferences of their target dispersers. Fruits consumed by migrating birds are often rich in lipids that provide the high energy fats necessary for long distance flight. Fruits eaten by primates tend to be high in sugars that provide quick energy for active animals, while fruits consumed by specialized frugivores may contain specific nutrients or compounds that these animals require for health and reproduction. Seasonal fruit production creates temporal niches that support diverse communities of disperser animals while ensuring that plants have access to dispersal services throughout their reproductive periods. Early season fruits provide resources when alternative foods may be scarce, while late season fruits help sustain disperser populations through periods of natural food shortage. This temporal partitioning reduces competition between plant species for disperser services while maintaining year-round partnerships that benefit entire ecological communities. The coevolution of plants and their seed dispersers has led to remarkable examples of mutual adaptation. The gape width of bird beaks has evolved to match the size of preferred fruits, while fruit sizes have been shaped by the feeding capabilities of target dispersers. The gut retention time of seeds in different animals has influenced seed size and protective coating thickness, creating precisely calibrated relationships between seed traits and disperser physiology. Climate change poses new challenges to seed dispersal relationships by altering the timing of fruit production and the distribution and behavior of disperser animals. Shifts in temperature and precipitation patterns can disrupt the careful synchronization between fruit ripening and disperser activity, while changes in habitat distribution may separate plants from their traditional dispersal partners. Understanding and protecting these relationships becomes increasingly important as species attempt to track suitable climatic conditions across changing landscapes. The loss of large-bodied disperser animals represents a particular threat to seed dispersal networks. Many large-seeded plants evolved in partnership with megafauna that are now extinct or greatly reduced in abundance. Without their original dispersal partners, these plants face significant challenges in establishing new populations and maintaining genetic connectivity across fragmented landscapes. Conservation efforts increasingly focus on protecting both plants and their disperser partners as integrated systems rather than individual species. Urban environments create novel challenges and opportunities for seed dispersal relationships. City parks and gardens provide resources for many frugivorous animals while creating new dispersal networks that may connect urban green spaces with surrounding natural areas. However, urban environments also introduce barriers to dispersal and expose disperser animals to risks like window collisions and traffic mortality that can disrupt traditional dispersal patterns. As night falls across a tropical forest, diurnal fruit dispersers give way to their nocturnal counterparts. Fruit bats emerge from their roosts to visit flowering and fruiting trees, providing dispersal services for plants that have evolved to attract these flying mammals through strong aromas and fruits that ripen under cover of darkness. These nighttime dispersal networks complement daytime activities, ensuring that seed dispersal continues around the clock in the world's most diverse ecosystems. The study of seed dispersal reveals the profound interconnectedness of plant and animal communities. Every fruit consumed, every seed transported, every successful establishment of a new plant individual represents the continuation of partnerships that have sustained forest diversity for millions of years. In the simple act of a bird eating a berry and flying to a new location, we witness one of nature's most fundamental forms of cooperation, a relationship that has shaped the distribution of life across our planet and continues to influence the structure and function of ecosystems worldwide. The gentle rustle of leaves as a monkey moves through the forest canopy, carrying within its stomach the seeds of tomorrow's trees, reminds us that the future of forests depends not on any single species, but on the intricate web of relationships that connect all living things in networks of mutual dependence and shared benefit. Beneath the forest floor, where sunlight never penetrates and silence reigns supreme, an intricate network of communication and cooperation spans for miles in every direction. Thread-like fungal filaments, finer than spider silk and more extensive than any human-built infrastructure, weave through soil particles and connect the roots of trees in partnerships that have sustained forests for over 400 million years. This is the mycorrhizal network, nature's original internet, where information, nutrients, and chemical signals flow continuously between partners in one of Earth's most ancient and essential cooperative relationships. The word mycorrhiza, meaning fungus root, barely hints at the sophistication of these underground alliances. Far from simple parasitic or commensal relationships, mycorrhizal partnerships represent true mutualism, where both plant and fungal partners derive essential benefits that neither could achieve alone. The fungi gain access to the sugars and carbon compounds that only photosynthetic plants can produce, while the plants receive vastly expanded access to soil nutrients and water that their own root systems could never obtain independently. The scale of these networks defies imagination. A single handful of forest soil may contain miles of fungal filaments, while the mycorrhizal network associated with a mature tree can extend for acres in every direction. Scientists estimate that more than 90% of all plant species form mycorrhizal partnerships, making these relationships one of the most ubiquitous forms of cooperation in terrestrial ecosystems. The fungal partners in these relationships represent some of the largest organisms on Earth, with individual fungal networks documented to span thousands of acres and persist for centuries. The anatomy of mycorrhizal partnerships reveals elegant solutions to the fundamental challenges facing both plants and fungi. Plant roots, while effective at anchoring trees and accessing nearby resources, are relatively thick and cannot penetrate the smallest soil spaces where many nutrients reside. Fungal hyphae, by contrast, are microscopically thin and can thread through soil pores too small for root penetration, accessing nutrient-rich microsites throughout the soil profile. The surface area provided by these fungal networks can increase a plant's nutrient-absorbing capacity by hundreds or even thousands of times compared to roots alone. The exchange mechanisms underlying mycorrhizal partnerships operate through specialized structures that facilitate efficient resource transfer between partners. In ectomycorrhizal relationships, fungal sheaths surround root tips, while hyphal networks extend outward into the soil, creating living bridges between plant and fungal metabolism. In endomycorrhizal associations, fungal structures actually penetrate root cell walls, creating intimate cellular connections that allow direct exchange of molecules between plant and fungal cytoplasm. The biochemistry of these exchanges involves sophisticated molecular recognition systems that ensure cooperation rather than exploitation. Plants can regulate the flow of carbon to their fungal partners based on the nutrients they receive in return, creating market-like dynamics where productive partnerships are rewarded while less beneficial associations are diminished. Fungi, meanwhile, can adjust their nutrient provisioning based on the carbon supply from their plant partners, creating feedback loops that maintain balanced exchanges over time. Seasonal rhythms govern many aspects of mycorrhizal partnerships, reflecting the changing needs of both partners throughout the year. During spring growth periods when trees are producing new leaves and wood, carbon flow to fungal partners may decrease as plants invest heavily in above-ground structures. In autumn, as trees prepare for winter dormancy, increased carbon allocation to mycorrhizal networks helps fungi maintain soil nutrient absorption during the cold months when root activity declines. The diversity of mycorrhizal relationships reflects the evolutionary creativity that has shaped these partnerships across different ecosystems and plant families. Ectomycorrhizal associations, common in temperate and boreal forests, involve fungi that form external mantles around root tips while extending exploration networks deep into surrounding soil. These partnerships often involve trees like oaks, pines, and birches working with fungi that produce the familiar mushrooms we see scattered across forest floors. Arbuscular mycorrhizal relationships, found in grasslands, tropical forests, and agricultural systems, involve fungi that penetrate root tissues and form intricate branching structures called arbuscules within plant cells. These intimate associations facilitate rapid and efficient nutrient exchange while allowing plants to maintain control over the partnership through their ability to regulate fungal access to root tissues. The communication capabilities of mycorrhizal networks extend far beyond simple nutrient exchange. Research has revealed that these fungal networks can transmit chemical signals between connected plants, allowing forest communities to coordinate responses to environmental challenges When one tree faces attack by insects or pathogens, it may release chemical alarm signals that travel through the mycorrhizal network to warn neighboring plants, which can then activate their own defensive mechanisms in preparation for potential threats. This chemical communication system appears to facilitate complex information sharing that benefits entire forest communities. Young seedlings connected to mature trees through mycorrhizal networks may receive not only nutrients, but also chemical signals that help them recognize and respond appropriately to various environmental conditions. Mother trees, as researchers have begun calling the largest and oldest individuals in forest stands, may actually nurture their offspring through mycorrhizal connections, providing carbon and nutrients that improve seedling survival and growth. The role of mycorrhizal networks in forest resilience has become increasingly apparent as researchers study how forests respond to disturbances like drought, fire, and climate change. Trees connected by extensive mycorrhizal networks often show greater resistance to environmental stress than isolated individuals, suggesting that the mutual support provided by these partnerships enhances the stability of entire forest ecosystems. When some trees in a network experience resource abundance while others face scarcity, the mycorrhizal connections can facilitate resource sharing that helps the entire community weather difficult periods. Soil chemistry is fundamentally altered by mycorrhizal partnerships through the activities of both plant roots and fungal hyphae. Fungi release enzymes that break down complex organic compounds, making nutrients available not only to their plant partners, but to the entire soil microbial community. The hyphal networks also create soil structure by binding particles together with sticky compounds, improving water retention and root penetration while reducing erosion and nutrient leaching. The carbon storage capabilities of mycorrhizal networks represent a crucial but often overlooked component of global carbon cycles. Fungi store substantial amounts of carbon in their biomass and in the soil organic compounds they produce, effectively sequestering atmospheric carbon dioxide that was originally fixed by their photosynthetic plant partners. Some estimates suggest that mycorrhizal fungi may store as much carbon below ground as is contained in all above ground plant biomass, making these partnerships critical components of Earth's climate regulation systems. Different soil types and environmental conditions favor different types of mycorrhizal partnerships, creating geographic patterns in the distribution of these relationships that influence global forest composition. Nutrient-poor soils often support extensive ectomycorrhizal networks that excel at extracting nutrients from organic matter, while fertile soils may favor arbuscular mycorrhizal associations that are more efficient at rapid nutrient uptake during active growth periods. The evolutionary history of mycorrhizal partnerships extends back to the earliest terrestrial ecosystems, when the first plants were colonizing land environments that lacked the complex soil communities we see today. Fossil evidence suggests that the earliest land plants formed mycorrhizal associations, and these partnerships may have been essential for plant colonization of terrestrial environments. The fungi provided early plants with access to mineral nutrients from rocky substrates, while the plants provided fungi with organic carbon compounds that were otherwise scarce in early terrestrial ecosystems. Agricultural systems have largely disrupted the natural mycorrhizal networks that once sustained plant communities, but growing recognition of their importance is leading to new approaches in sustainable farming. Reduced tillage practices help preserve fungal networks, while diverse crop rotations and cover cropping can maintain mycorrhizal diversity and function Some farmers are now deliberately inoculating their soils with beneficial mycorrhizal fungi to restore these partnerships and reduce dependence on synthetic fertilizers. Climate change poses both challenges and opportunities for mycorrhizal partnerships. Rising temperatures and altered precipitation patterns may disrupt established fungal communities while creating opportunities for new partnerships to develop. The enhanced carbon dioxide levels associated with climate change may increase plant carbon production, potentially strengthening mycorrhizal partnerships if adequate soil nutrients are available to support increased fungal activity. Urban environments present unique challenges for mycorrhizal partnerships through soil compaction, pollution, and fragmentation of natural fungal networks. However, research is revealing that mycorrhizal relationships can help urban plants cope with environmental stresses like air pollution and drought, making the restoration of these partnerships an important consideration in urban forestry and landscape management. The study of mycorrhizal networks requires sophisticated techniques that can track the flow of nutrients and information through underground fungal connections. Isotopic tracers allow researchers to follow the movement of carbon, nitrogen, and phosphorus between plants and their fungal partners. Molecular techniques can identify the specific fungal species involved in different partnerships and track changes in fungal community composition over time. As darkness settles over the forest canopy, the underground economy continues its ancient rhythms. Fungal networks pulse with the flow of nutrients harvested from soil organic matter, while plant roots provide steady streams of freshly manufactured sugars to their subterranean partners. This hidden commerce, invisible to surface observers but essential to forest function, represents one of nature's most successful cooperative enterprises. The mycorrhizal web beneath our feet connects individual trees into forest communities, individual forests into regional ecosystems, and individual ecosystems into the global networks that sustain terrestrial life. In the quiet chemistry of root and fungus, in the patient exchange of carbon for nutrients, in the generous sharing of resources between neighbors, we find a model of cooperation that has sustained life on land for hundreds of millions of years. These underground partnerships remind us that cooperation often occurs beyond our direct observation, in the hidden places where life's most essential work is accomplished through patient mutual aid and shared benefit. In a sun-dappled meadow where crimson clover blooms stretch toward the morning light, an invisible transformation is taking place that forms the foundation of life on Earth. Deep within the root systems of these legume plants, microscopic bacterial partners are performing a feat of biological alchemy that no other organisms can accomplish. They are capturing nitrogen from the air itself and converting it into forms that plants can use to build proteins, nucleic acids, and all the other nitrogen-containing molecules essential for life. This partnership between plants and nitrogen-fixing bacteria represents one of the most crucial cooperative relationships in terrestrial ecosystems. Nitrogen, while abundant in Earth's atmosphere, exists primarily as molecular nitrogen gas that plants cannot directly utilize. The specialized bacteria living within root nodules of leguminous plants bridge this gap, using complex enzyme systems to break the strong triple bonds of atmospheric nitrogen and incorporate it into organic compounds that fuel the growth and reproduction of their plant hosts. The roots of clover, alfalfa, soybeans, and countless other legume species are dotted with small, rounded structures called nodules that house millions of these bacterial partners. Each nodule represents a sophisticated biological factory where plant and bacterial metabolism are intimately integrated. The plant provides its bacterial partners with carbohydrates, amino acids, and a carefully controlled environment, while the bacteria supply the plant with fixed nitrogen that would otherwise be unavailable from soil sources alone. The formation of root nodules involves an intricate molecular dialogue between plant and bacterial partners that rivals the complexity of any diplomatic negotiation. When legume seeds germinate and their roots begin exploring the surrounding soil, they release specific chemical signals called flavonoids that attract compatible nitrogen fixing bacteria. The bacteria respond with their own molecular messages called nod factors that trigger dramatic changes in root development and prepare the plant for bacterial colonization. The specificity of this recognition system ensures that plants form partnerships only with bacterial strains that can provide effective nitrogen fixation services. Different legume species often partner with different bacterial species, creating a diverse array of plant bacterial combinations that have evolved together over millions of years. Alfalfa typically partners with Cynorhizobium meliloti, while soybeans form relationships with Bradyrhizobium japonicum, and each partnership represents a precisely calibrated alliance optimized for the specific needs of both partners. The biochemistry of nitrogen fixation requires enormous amounts of energy and sophisticated cellular machinery that showcases the remarkable capabilities of bacterial metabolism. The enzyme complex responsible for nitrogen fixation, called nitrogenase, is one of the most energy expensive biological processes known, requiring 16 molecules of ATP for every molecule of nitrogen gas converted to ammonia. This massive energy requirement explains why nitrogen fixing bacteria depend so heavily on their plant partners for carbohydrate supplies. The cellular environment within root nodules is carefully engineered to support nitrogen fixation while protecting the delicate enzyme systems from damage. Nitrogenase is extremely sensitive to oxygen, which can irreversibly destroy its catalytic activity. Plant cells within nodules produce specialized proteins called leghemoglobins that bind oxygen and maintain the low oxygen conditions necessary for effective nitrogen fixation. These proteins give active nodules their characteristic pink or red coloration, a visible sign of the partnership's productivity. The regulation of nitrogen fixation demonstrates sophisticated feedback mechanisms that prevent waste and optimize resource allocation for both partners. When soil nitrogen levels are high, plants reduce their investment in bacterial partners and rely more heavily on direct nutrient uptake from the soil. When nitrogen becomes scarce, the partnership intensifies, with plants providing more resources to their bacterial partners in exchange for increased nitrogen fixation services. Seasonal cycles create dynamic patterns in nitrogen fixing partnerships that reflect the changing needs of plant communities throughout the growing season. In temperate regions, nitrogen fixation typically peaks during the rapid growth periods of late spring and early summer, when plants are actively producing new tissues with high nitrogen requirements. As autumn approaches and plants prepare for dormancy, nitrogen fixation gradually declines, allowing bacterial partners to conserve energy for overwintering survival. The ecological impact of nitrogen-fixing partnerships extends far beyond the individual plants involved in these relationships. Fixed nitrogen gradually accumulates in soil organic matter as roots and nodules decompose, creating pools of available nitrogen that benefit entire plant communities. This process, called biological nitrogen fixation, is responsible for adding millions of tons of nitrogen to terrestrial ecosystems annually, far exceeding the contributions from industrial nitrogen fixation or atmospheric deposition. Agricultural systems have long recognized and exploited the benefits of nitrogen fixing partnerships through crop rotation practices that alternate nitrogen demanding crops with nitrogen fixing legumes. A field planted with corn or wheat one year might be planted with soybeans or clover the next, allowing the legume crop to replenish soil nitrogen depleted by the previous crop. This practice reduces the need for synthetic nitrogen fertilizers while maintaining soil fertility and supporting sustainable agricultural production. The diversity of nitrogen fixing partnerships encompasses relationships far beyond the familiar legume bacteria associations. In forest ecosystems, alder trees form partnerships with Frankia bacteria that allow them to colonize nitrogen-poor soils where other trees cannot establish. These actinorhizal relationships enable alders to serve as pioneer species in disturbed areas, gradually building soil nitrogen levels that eventually support more diverse forest communities. Aquatic ecosystems host their own specialized nitrogen-fixing partnerships, including relationships between cyanobacteria and various aquatic plants. Water ferns in the genus Azolla maintain chambers within their leaves that house nitrogen-fixing cyanobacteria, creating floating partnerships that can contribute substantial amounts of fixed nitrogen to wetland and rice paddy ecosystems. These aquatic partnerships demonstrate the fundamental importance of biological nitrogen fixation across diverse habitats. The global nitrogen cycle depends heavily on biological nitrogen fixation to balance the continuous loss of nitrogen through denitrification and other processes that return nitrogen to the atmosphere. Without the ongoing activities of nitrogen-fixing bacteria and their plant partners, terrestrial ecosystems would gradually become nitrogen-limited, constraining plant growth and reducing the productivity of entire ecological communities. Climate change is creating new challenges and opportunities for nitrogen-fixing partnerships through its effects on temperature, precipitation, and atmospheric carbon dioxide levels. Higher temperatures may enhance nitrogen fixation rates in some regions while creating stress in others, and altered precipitation patterns could affect the soil conditions necessary for effective root nodule formation and function. Rising carbon dioxide levels may increase plant carbon production, potentially strengthening partnerships by providing bacterial partners with more abundant carbohydrate supplies. The molecular mechanisms underlying nitrogen fixation continue to inspire research into sustainable alternatives to energy-intensive industrial nitrogen production. Understanding how bacterial nitrogenase operates under mild biological conditions may lead to new catalytic processes that could reduce the environmental costs of synthetic fertilizer production while maintaining agricultural productivity. Soil management practices in both agricultural and natural systems increasingly recognize the importance of supporting nitrogen-fixing partnerships through appropriate environmental conditions. Adequate soil phosphorus, proper pH levels, and protection from excessive disturbance all contribute to successful nodulation and effective nitrogen fixation. Conservation practices that maintain diverse plant communities, including native legumes, help preserve the full spectrum of nitrogen-fixing partnerships that support ecosystem health. The evolution of nitrogen-fixing partnerships required remarkable coordination between very different forms of life, with plants developing the cellular machinery necessary to house bacterial partners, while bacteria evolved the metabolic pathways necessary to provide nitrogen in forms that plants could utilize. This coevolutionary process has produced some of the most tightly integrated partnerships in biology, where the success of each partner depends completely on the contributions of the other. Urban environments present unique challenges for nitrogen fixing partnerships through soil contamination, compaction, and altered nutrient cycles, but these relationships also offer opportunities for sustainable landscape management in developed areas. Native leguminous trees and shrubs can provide nitrogen inputs that support urban forest health while reducing dependence on synthetic fertilizers that can contribute to water pollution. The study of nitrogen fixing partnerships has revealed fundamental principles about resource exchange, partner recognition, and cooperative evolution that apply broadly across biological systems. The molecular signals that initiate these partnerships, the cellular structures that house them, and the regulatory mechanisms that maintain them provide models for understanding cooperation in other contexts. As evening settles over agricultural landscapes and natural ecosystems, the quiet work of nitrogen fixation continues in millions of root nodules throughout the soil. Bacterial partners process atmospheric nitrogen through ancient metabolic pathways, while plant partners provide the energy and cellular environment necessary for this essential transformation. This hidden partnership, invisible to casual observation but fundamental to life's abundance, demonstrates that some of nature's most important cooperative relationships occur in the microscopic realm where chemistry and biology intersect. The relationship between legumes and their bacterial partners reminds us that cooperation often requires intimate integration, where partners become so interdependent that their individual success becomes inseparable from their mutual success. In the quiet chemistry of nitrogen fixation, we find a model of partnership that sustains the productivity of both natural and agricultural ecosystems, while demonstrating the remarkable possibilities that emerge when different forms of life learn to work together toward common goals. On the weathered granite face of a mountain cliff, where few other organisms dare to venture, a pale green crust spreads slowly across the rock surface. To the casual observer, this might appear to be simple moss or algae clinging to stone, but closer examination reveals one of nature's most remarkable partnerships. This is a lichen, not a single organism at all, but a living collaboration between fungal and photosynthetic partners, so intimate that they function as a unified being, creating something entirely new from their alliance. The partnership that creates lichens represents an extreme form of cooperation where the boundaries between individual organisms become blurred. Unlike the temporary associations between flowers and their pollinators, or even the long-term relationships between plants and mycorrhizal fungi, lichen partnerships achieve such complete integration that the resulting composite organism looks, functions, and reproduces as a single entity. This biological fusion has enabled lichens to colonize some of Earth's most challenging environments, from Arctic tundra to desert rocks, demonstrating the extraordinary possibilities that emerge when cooperation reaches its ultimate expression. The anatomy of a lichen reveals the elegant architecture of integrated partnership. The fungal partner, typically an ascomycete, provides the structural framework that gives the lichen its shape and anchors it to its substrate. These fungal filaments create a protective matrix that shields the partnership from environmental extremes while providing mechanical support and water storage capacity. Within this fungal framework, the photosynthetic partner, usually a green alga or cyanobacterium, is carefully positioned to maximize light capture while remaining protected from desiccation and temperature fluctuations. The metabolic integration of lichen partnerships achieves remarkable efficiency in resource utilization. The photosynthetic partner converts sunlight, carbon dioxide, and water into organic compounds that fuel both partners' growth and maintenance. The fungal partner provides mineral nutrients extracted from the substrate, creates the physical structure that optimizes light exposure, and manages water relations that allow photosynthesis to continue even under harsh environmental conditions. This division of labor allows lichens to thrive in environments where neither partner could survive alone. The diversity of lichen forms reflects the creative potential of fungal algal partnerships under different environmental conditions. Crustose lichens spread as thin crusts across rock surfaces, maximizing adhesion and minimizing exposure to drying winds. Foliose lichens develop leaf-like structures that increase surface area for both light capture and gas exchange while remaining firmly attached to their substrates. Fruticose lichens grow upright or hanging, creating three-dimensional structures that can capture moisture from fog and rain while accessing light in competitive environments. The reproductive strategies of lichens demonstrate sophisticated coordination between partners that ensures the continuation of their integrated lifestyle. Some lichens reproduce through fragmentation, where pieces of the composite organism break off and establish new colonies, automatically maintaining the partnership in each fragment. Others produce specialized structures called sordia, tiny packages that contain both fungal and algal cells, wrapped together in a dispersible bundle that can colonize new substrates while preserving the essential partnership. The ecological roles of lichens extend far beyond their individual survival to encompass fundamental ecosystem processes that support entire biological communities. As pioneer species, lichens are often the first organisms to colonize bare rock surfaces, beginning the slow process of soil formation that eventually enables other plants to establish. Through their metabolic activities, lichens gradually break down rock substrates, releasing mineral nutrients and creating organic matter that accumulates to form primitive soils. The biochemistry of rock weathering by lichens involves the production of organic acids that slowly dissolve mineral components of rock substrates. These acids, including oxalic acid, citric acid, and various phenolic compounds, are secreted by the fungal partner and concentrated at the lichen rock interface. Over time, scales measured in decades or centuries, this chemical weathering creates shallow depressions and crevices that collect organic matter and provide establishment sites for mosses, small flowering plants, and eventually larger vegetation. In Arctic and alpine environments, lichens form the foundation of entire food webs, providing essential nutrition for herbivores that few other organisms could support in these harsh conditions. Reindeer and caribou depend heavily on lichen forage during winter months when other food sources are unavailable or inaccessible under snow. The slow growth rates of lichens, typically measured in millimeters per year, make these ecosystems particularly vulnerable to overgrazing and environmental disturbances that can require decades or centuries for recovery. The sensitivity of lichens to air pollution has made them valuable indicators of environmental quality, particularly for sulfur dioxide and heavy metal contamination. The absence of protective waxy cuticles that characterize vascular plants makes lichens highly susceptible to airborne pollutants, which they absorb directly from the atmosphere. Changes in lichen diversity and abundance often provide early warning signs of air quality degradation that precede more obvious effects on other organisms. Different lichen species exhibit varying tolerances to specific types of environmental stress, creating patterns of distribution that reflect local environmental conditions. Pollution sensitive species disappear first from contaminated areas, while more tolerant species may persist or even increase in abundance. This differential sensitivity has enabled scientists to develop lichen based biomonitoring systems that can detect and quantify air pollution levels with remarkable precision. The longevity of some lichen colonies rivals that of the longest lived trees, with individual colonies potentially persisting for thousands of years under favorable conditions. This extraordinary lifespan reflects the efficiency of their partnership in utilizing limited resources and their ability to survive extended periods of environmental stress through dormancy and reduced metabolic activity. During dry periods, lichens can lose most of their water content and enter a state of cryptobiotic suspension, resuming active metabolism within minutes of rewetting. The global distribution of lichens encompasses virtually every terrestrial environment on Earth, from tropical rainforests to polar regions, from sea level to high mountain peaks. This remarkable range reflects the adaptability of the fungal algal partnership and its ability to fine tune metabolic and structural characteristics to match local environmental conditions. Different combinations of fungal and algal partners have evolved to optimize performance under specific temperature, moisture, and light regimes. The study of lichen partnerships has revealed fundamental insights into the evolution of cooperation and the potential for unrelated organisms to achieve functional integration. The transition from free-living fungi and algae to integrated lichen partnerships required extensive coevolutionary changes in both partners, including modifications to cell wall chemistry, development of nutrient transport systems, and coordination of reproductive cycles. Modern molecular techniques have revolutionized our understanding of lichen partnerships by revealing that many lichens actually involve more than two partners. In addition to the primary fungal and algal components, many lichens harbor additional bacterial partners that contribute to nitrogen fixation, production of secondary metabolites, or other specialized functions. These discoveries suggest that lichen partnerships may be even more complex and integrated than previously recognized. The biochemical diversity of lichens has attracted attention from researchers seeking new pharmaceuticals and industrial compounds. Many lichens produce unique secondary metabolites that serve protective or competitive functions within the partnership. These compounds, which include antibiotics, antiviral agents, and UV screening pigments, represent a largely untapped reservoir of potentially useful natural products. Climate change poses complex challenges for lichen partnerships through its effects on temperature, precipitation patterns, and atmospheric composition. Some lichen species may benefit from increased carbon dioxide levels and longer growing seasons, while others may suffer from increased temperatures, altered precipitation patterns, or changes in the frequency of freeze-thaw cycles. The slow growth and long generation times of many lichens may limit their ability to adapt rapidly to changing environmental conditions. Conservation of lichen diversity requires protection of the specific microhabitats and environmental conditions that support different species. Many rare lichen species depend on old growth forests, undisturbed rock formations, or other specialized habitats that are increasingly threatened by human development. The restoration of lichen communities in disturbed areas is challenging due to their slow growth rates and specific substrate requirements. Urban environments present both challenges and opportunities for lichen partnerships. Air pollution and habitat fragmentation reduce lichen diversity in many urban areas, but some species adapt to urban conditions and may even benefit from reduced competition and novel substrates provided by buildings and infrastructure. Urban lichen communities often differ significantly from those in natural areas, reflecting the selective effects of urban environmental conditions. The cultural significance of lichens varies among different human societies, with some indigenous peoples using lichens for food, medicine, dyes, and other purposes. The traditional ecological knowledge associated with lichen use provides valuable insights into the biology and ecology of these partnerships while demonstrating their importance to human communities in environments where few other resources are available. As night settles over landscapes where lichens slowly pursue their ancient partnership, the quiet chemistry of cooperation continues in countless locations around the globe. On mountain cliffs and forest trees, on Arctic soils and desert rocks, fungal and algal partners maintain their integrated existence through patient resource sharing and mutual care. These composite organisms, products of evolution's most creative experiments in cooperation, remind us that the boundaries between self and other, between individual and community, are often more fluid than we might imagine. In the slow growth of lichens across stone surfaces, in their patient colonization of the Earth's most challenging environments, we find a model of partnership that transcends the limitations of individual existence to create something greater than the sum of its parts. In the humid canopy of a Costa Rican rainforest, a young acacia tree extends hollow thorns toward the morning light, each swollen spine containing a perfectly formed chamber that will soon house a colony of fierce protectors. Within days, winged queens of Pseudomyrmex ants will discover these botanical apartments, establishing colonies that will transform the tree into a living fortress. In exchange for shelter and food, the ants will patrol every branch and leaf, attacking any creature that dares to threaten their plant partner in one of nature's most militaristic cooperative relationships. The partnership between acacia trees and their ant guardians represents defensive cooperation at its most dramatic, where plants actively recruit animal bodyguards to protect them from the countless herbivores that would otherwise strip their leaves and destroy their growing tissues. This relationship, known as defensive mutualism, has evolved independently in tropical regions around the world. Wherever the constant pressure from plant-eating insects has created strong evolutionary incentives for plants to seek protection through partnership. The hollow thorns of swollen thorn acacias are architectural marvels specifically designed to house ant colonies. Unlike the solid defensive spines of most acacia species, these modified thorns develop hollow chambers with thin walls that ants can easily chew through to create entrance holes. The size and internal structure of these thorns are precisely calibrated to accommodate ant colonies while providing insulation from temperature extremes and protection from the frequent tropical rainstorms that could otherwise flood ant nests. The relationship extends far beyond simple housing arrangements. Acacia trees provide their ant partners with two specialized food sources that meet all their nutritional needs, eliminating the necessity for the ants to forage elsewhere. At the tips of their leaflets, acacias produce small, protein-rich structures called Beltian bodies that serve no function for the plant beyond feeding their ant guardians. These golden-colored food packages are harvested regularly by worker ants and provide essential amino acids and fats necessary for colony growth and reproduction. The second food source takes the form of nectar produced by specialized glands called extrafloral nectaries located at the base of leaf stems. Unlike the nectar produced by flowers to attract pollinators, this foliar nectar serves purely to nourish the plant's defensive partners. Rich in sugars and containing various amino acids, this nectar provides the quick energy that ant workers need for their constant patrol duties, while helping to maintain the high activity levels necessary for effective plant defense. The behavioral ecology of guardian ants reveals sophisticated strategies that maximize their protective effectiveness while minimizing energy expenditure. Worker ants establish regular patrol routes along branches and stems, maintaining territories that they defend aggressively against intruders. These patrols operate around the clock, with different shifts of workers ensuring continuous surveillance of their plant partners' vulnerable tissues. When threats are detected, the response is swift and coordinated. Herbivorous insects that land on acacia foliage are immediately attacked by resident ants, which swarm the intruders and drive them away through biting and stinging. The ants' response is so rapid and aggressive that most insects learn to avoid acacia trees entirely, creating an exclusion zone around protected plants that benefits not only the acacias, but also neighboring vegetation that receives reduced herbivore pressure. The chemical warfare capabilities of guardian ants add another dimension to plant protection. Many ant species inject venoms through their stings that can kill or disable small arthropods while causing painful welts on larger animals. Some ants also spray formic acid or other deterrent chemicals that make protected plants thoroughly unpalatable to potential herbivores. These chemical defenses complement the ants' physical aggression and create multiple layers of protection for their plant partners. The protection provided by ant guardians extends beyond defense against herbivorous insects to include removal of competing vegetation. Worker ants regularly patrol the ground around their host trees, cutting down or girdling competing plants that might shade their partner or compete for soil nutrients. This vegetation management creates clearings around protected trees that reduce competition while providing the ants with additional nesting sites in the form of cleared soil areas. The evolutionary arms race between plants, their ant guardians, and the herbivores they defend against has produced remarkable adaptations on all sides. Some herbivorous insects have evolved behaviors that allow them to exploit ant-protected plants by timing their feeding activities to avoid ant patrols or by producing chemical compounds that mask their presence from ant detection. Systems, plants and ants in turn, have evolved counter-adaptations that maintain the effectiveness of their defensive partnership. The specificity of many ant-plant relationships reflects millions of years of coevolution between particular plant and ant lineages. Bull's horn acacias partner almost exclusively with Pseudomyrmex ferrugineus ants, while other acacia species form relationships with different ant species. This specificity ensures that both partners are precisely adapted to each other's needs and capabilities, maximizing the efficiency of their cooperative relationship. The reproductive strategies of guardian ants are intimately tied to the life cycles of their plant partners. Queen ants time their nuptial flights to coincide with the production of new acacia thorns, ensuring that newly mated queens can quickly locate suitable nest sites. The establishment of new ant colonies on young acacia trees creates partnerships that can persist for decades as both partners grow and mature together. Cecropia trees of Central and South America have evolved their own version of the ant plant partnership, providing specialized structures called Mullerian bodies that serve as ant food while maintaining hollow internodes that house Azteca ant colonies. These relationships demonstrate the convergent evolution of defensive mutualism, where similar ecological pressures have led to the independent development of comparable cooperative strategies in different plant families. The nutritional quality of plant-produced ant food reflects sophisticated understanding of ant dietary requirements. Beltian bodies contain essential amino acids in proportions that closely match the needs of growing ant larvae, while the lipid content provides energy-dense nutrition that supports the high activity levels of worker ants. This precision in nutritional provisioning suggests that plants have evolved detailed knowledge of their partner's biological needs. The seasonal dynamics of ant-plant relationships reveal complex patterns of resource allocation and defensive intensity. During dry seasons, when herbivore pressure may be reduced, some plants decrease their production of ant foods, allowing energy to be diverted to growth and reproduction. When rains return and herbivore activity increases, food production for ant partners intensifies, ensuring that defensive services are available when most needed. The impact of ant guardians on plant fitness extends beyond simple protection from herbivores to include effects on plant growth, reproduction, and survival. Trees with ant partners typically show faster growth rates, higher survival during droughts, and greater reproductive success compared to undefended individuals. These benefits justify the substantial energy investments that plants make in housing and feeding their ant guardians. Climate change poses new challenges for ant-plant partnerships through its effects on temperature, precipitation, and the seasonal timing of biological activities. Changes in rainfall patterns may affect the production of extrafloral nectar, while temperature increases could stress both plant and ant partners. Shifts in the timing of herbivore activity relative to ant life cycles could disrupt the synchronization necessary for effective plant defense. The study of ant-plant relationships has revealed important principles about the evolution of cooperation, including the conditions that favor the development of mutualistic partnerships and the mechanisms that prevent partners from cheating or exploiting each other. The ability of plants to withhold resources from ineffective ant colonies, combined with the ants' dependence on plant-provided food and shelter, creates strong incentives for both partners to maintain their cooperative behavior. Agricultural applications of ant-plant partnership principles are being explored as alternatives to pesticide-based crop protection strategies. Some coffee and cacao plantations maintain shade trees that support ant colonies, providing biological pest control services that reduce the need for synthetic insecticides. Understanding the requirements for successful ant-plant partnerships may lead to new approaches for sustainable agriculture in tropical regions. Urban environments present unique challenges for ant-plant relationships through habitat fragmentation and the introduction of invasive ant species that may disrupt established partnerships. Conservation of these relationships requires protection of the specific environmental conditions that support both partners, including the maintenance of habitat corridors that allow for the movement of founding ant queens between suitable plant hosts. As darkness falls over tropical forests, where ant-plant partnerships have flourished for millions of years, the vigilant guardians continue their patrols through the canopy. Worker ants move along familiar routes, their chemical sensors alert for signs of threat, their mandibles ready to defend their plant partners against any creature that would do them harm. In the quiet rustle of leaves and the nearly imperceptible footsteps of tiny guardians, we witness one of nature's most successful examples of interspecies cooperation, where protection and provision create bonds that transform individuals into inseparable partners. These botanical fortresses remind us that cooperation in nature often requires significant investments from both partners, but that the rewards of successful partnership can provide benefits that far exceed the costs of individual effort. In the crystalline waters of a coral reef, where shafts of tropical sunlight create dancing patterns on the sandy bottom, a magnificent sea turtle glides slowly toward a familiar coral outcropping. As the ancient reptile settles onto the reef, tiny cleaner fish emerge from their crevices and begin their delicate work, removing algae and parasites from the turtle's shell and skin. Nearby, a bright orange clownfish darts playfully among the swaying tentacles of its sea anemone host, immune to the stings that would paralyze other fish of similar size. These scenes of underwater cooperation reveal a marine world where partnership and mutual aid are as essential as the tides themselves. The ocean, covering more than 70% of Earth's surface, hosts cooperative relationships of extraordinary diversity and complexity, from the microscopic partnerships between coral polyps and their algal symbionts to the vast networks of cooperation that sustain entire reef ecosystems. Marine environments demonstrate that collaboration is a fundamental organizing principle of life beneath the waves. The three-dimensional nature of aquatic habitats, the constant movement of water masses, and the unique challenges of life in saltwater have fostered cooperative strategies that often have no parallel in terrestrial ecosystems. Coral reefs themselves represent perhaps the most spectacular example of marine cooperation, built entirely through the partnership between coral animals and microscopic algae called zooanthellae. Each coral polyp, no larger than a pencil eraser, houses millions of these photosynthetic partners within its tissues. The algae capture sunlight and convert it into organic compounds that provide up to 90% of the coral's nutritional needs, while the coral provides its algal partners with protection, nutrients, and the carbon dioxide they need for photosynthesis. This fundamental partnership enables corals to build the massive limestone structures that create reef environments supporting nearly a quarter of all marine species. The slow, patient accumulation of calcium carbonate skeletons over thousands of years creates the complex three-dimensional habitat that serves as foundation for countless other cooperative relationships. Without the initial partnership between coral and algae, these underwater cities of cooperation could not exist. The relationship between clownfish and sea anemones demonstrates how marine partnerships can provide mutual benefits while creating some of the ocean's most iconic imagery. The anemone's stinging tentacles, armed with specialized cells called nematocysts that inject paralyzing toxins, create an impenetrable fortress that most fish must avoid. Clownfish, however, have evolved special mucus coatings that protect them from the anemone's stings, allowing them to live safely among the deadly tentacles. The clownfish gains not only protection from predators, but also access to food scraps from the anemone's meals and a safe place to lay their eggs. The anemone benefits from the clownfish's aggressive territorial behavior, which drives away fish that might otherwise feed on the anemone's tentacles. The clownfish also provides cleaning services, removing parasites and dead tissue from their host, while their constant movement helps circulate water through the anemone's body cavity, improving gas exchange and waste removal. Cleaning stations scattered throughout coral reef systems create hubs of cooperative activity where multiple species converge for health maintenance services. These underwater clinics operate with remarkable organization as large fish queue patiently for their turn to receive grooming services from small, cleaner fish and shrimp. The predictable locations and timing of cleaning services create opportunities for numerous other cooperative relationships to develop around these centers of activity. The cleaner wrasse, a small fish with distinctive blue and black striping, has evolved specialized feeding behaviors that make it an extremely effective partner in these relationships. Its pointed snout and precise movements allow it to remove parasites from sensitive areas like gill chambers and mouth cavities without causing harm to its clients. The wrasse has even developed the ability to enter the mouths of large predatory fish, trusting in the temporary truce that cleaning relationships establish. Cleaner shrimp add another dimension to reef cleaning services, using their specialized claws to remove parasites and dead tissue with remarkable precision. These small crustaceans often establish cleaning stations within the protective embrace of sea anemones, creating multi-species partnerships where the shrimp receives protection while the anemone benefits from increased fish traffic that occasionally results in prey capture. The partnership between remoras and their large host fish demonstrates how cooperation can enable access to resources that would otherwise be unavailable. These specialized fish have evolved modified dorsal fins that function as suction cups, allowing them to attach securely to sharks, rays, sea turtles, and marine mammals. The remora gains transportation to food-rich areas and protection from predators, while providing cleaning services to its host by removing parasites and dead tissue. The relationship extends beyond simple hitchhiking, as remoras often feed on scraps from their host's meals and may help their partners locate food by alerting them to potential prey. Some marine biologists suggest that remoras may also provide early warning of approaching predators or competitors through their behavior, adding a security dimension to the cleaning services they provide. Schools of fish that include multiple species demonstrate how temporary cooperative relationships can provide benefits that extend beyond the immediate participants. Mixed species schools often form around feeding opportunities or in response to predator threats, combining the different sensory capabilities and behavioral repertoires of various fish species. Some species excel at detecting predators, others at locating food sources, and still others at escape maneuvers, creating temporary partnerships that enhance survival for all participants. The deep ocean, far from the sunlit coral reefs, hosts its own remarkable cooperative relationships, adapted to the challenges of life in perpetual darkness. At hydrothermal vents scattered along mid-ocean ridges, entire ecosystems depend on partnerships between animals and chemosynthetic bacteria that can extract energy from the hydrogen sulfide and other chemicals spewing from these underwater geysers. Giant tube worms, some reaching lengths of 8 feet, have no digestive systems of their own, but instead house vast colonies of chemosynthetic bacteria within specialized organs called trophosome. The bacteria convert toxic hydrogen sulfide into organic compounds that nourish their worm hosts, while the worms provide the bacteria with a stable environment and access to the chemicals they need for their metabolism. This partnership enables complex communities to thrive in environments that were once thought to be too harsh to support life. The bioluminescent displays that characterize many deep sea creatures often serve cooperative functions, with different species coordinating their light production to create complex signaling networks. Some deep sea fish maintain partnerships with luminous bacteria housed in specialized light organs, providing the bacteria with nutrients and a stable environment in exchange for controllable bioluminescence that can be used for communication, prey attraction, or predator deterrence. Whale fall communities represent temporary but highly productive cooperative ecosystems that develop around the carcasses of dead whales that sink to the ocean floor. These massive food sources support complex succession communities where different species partnerships dominate at different stages of decomposition. Early colonizers include hagfish and sleeper sharks that consume soft tissues, followed by polychaete worms and crustaceans that work through remaining flesh, and finally bacteria that break down lipids in whale bones, supporting communities that may persist for decades. The great migrations of marine animals create moving networks of cooperation that span entire ocean basins. Humpback whales coordinate their feeding behaviors in Alaskan waters, working together to create bubble nets that concentrate krill into dense patches that can be efficiently harvested. These cooperative feeding strategies allow individual whales to access far more food than they could obtain through solitary hunting efforts. Sea turtle migrations connect cooperative relationships across thousands of miles of ocean as these ancient reptiles move between feeding areas, mating grounds, and nesting beaches. The remoras and cleaner fish that accompany migrating turtles provide mobile cleaning services while gaining access to diverse feeding opportunities across different marine environments. The seasonal dynamics of marine cooperation reflect the complex cycles of productivity that characterize ocean ecosystems. In polar waters, the brief summer blooms of phytoplankton create temporary abundance that supports intensive cooperative relationships between different species of whales, seabirds, and fish. These partnerships may last only a few months, but are essential for the survival and reproduction of species that must endure long periods of scarcity. Climate change and ocean acidification pose significant threats to marine cooperative relationships through their effects on water temperature, chemistry, and food web dynamics. Coral bleaching events caused by temperature stress that disrupts the partnership between corals and their algal symbionts demonstrate how environmental changes can break down cooperative relationships and cascade through entire ecosystems. The conservation of marine cooperative relationships requires protection not only of individual species, but of the ecological processes and environmental conditions that make these partnerships possible. Marine protected areas that encompass cleaning stations, spawning aggregations, and migration corridors help preserve the spatial and temporal patterns that many cooperative relationships depend upon. As moonlight filters down through ocean waters, the rhythm of marine cooperation continues in the darkness. Bioluminescent plankton coordinate their flashing to confuse predators, while deep sea creatures maintain partnerships with bacterial symbionts that provide the light necessary for communication and feeding. Night active cleaners emerge from their daytime refuges to service nocturnal clients, ensuring that the reef's health maintenance services continue around the clock. The vast blue wilderness of the ocean reminds us that cooperation transcends the boundaries between land and sea, creating networks of mutual aid and shared benefit that sustain life in Earth's most extensive habitat. In the gentle movements of a sea turtle receiving cleaning services, in the darting dance of clownfish among anemone tentacles, in the patient queuing of fish at cleaning stations, we find evidence that collaboration is truly a universal principle of life, as essential in the depths of the ocean as in the canopies of rainforests. Along the meandering course of a mountain stream, where clear water tumbles over moss covered stones and creates pools of liquid crystal, a diverse community of organisms has learned to work together in ways that transform individual struggle into collective success. In the shallow riffles, caddisfly larvae construct protective cases from tiny pebbles and organic debris, inadvertently creating habitat structure that benefits countless other small invertebrates. Downstream, a beaver dam spans the waterway, its patient engineering creating wetland habitat that supports species found nowhere else in the watershed. These freshwater partnerships demonstrate that cooperation flows as naturally through aquatic ecosystems as the water that sustains them. Freshwater environments, from the smallest mountain brooks to the mightiest rivers, host cooperative relationships that are shaped by the unique properties of flowing water and the seasonal rhythms that govern aquatic life. Unlike the relatively stable conditions of marine environments, freshwater systems experience dramatic fluctuations in temperature, water level, and chemical composition that create both challenges and opportunities for organisms seeking to form beneficial partnerships. The foundation of many freshwater cooperative relationships lies in the complex interactions between aquatic plants and the communities of bacteria and algae that colonize their surfaces. Submerged macrophytes like pondweeds and water milfoil provide substrate and nutrients for epiphytic communities that form living films across their leaf surfaces. These microbial gardens perform essential functions for their plant hosts, including nutrient cycling, protection from pathogens, and production of compounds that deter herbivores. The relationship extends beyond simple surface colonization to encompass sophisticated chemical communication between plants and their microbial partners. Many aquatic plants release specific compounds that attract beneficial bacteria while inhibiting harmful species, creating selective pressures that shape the composition of their associated microbial communities. In return, the bacteria produce growth promoting hormones, vitamins, and other compounds that enhance plant health and productivity. Stream ecosystems demonstrate remarkable examples of cooperative relationships between different functional groups of organisms. Leaf shredders, including various species of aquatic insects and crustaceans, break down fallen leaves and other organic debris that enters streams from surrounding terrestrial vegetation. This mechanical processing creates smaller particles and increases surface area for bacterial decomposition, accelerating nutrient release and making organic matter available to collector species that gather fine particulate matter from the water column. The collectors, in turn, process organic particles through their digestive systems, further breaking down complex compounds and releasing nutrients in forms that can be utilized by primary producers like algae and aquatic plants. This creates a cooperative processing chain where different species contribute specialized functions to the overall decomposition process, maximizing the efficiency of nutrient cycling within the stream ecosystem. Filter feeding organisms like freshwater mussels create cooperative relationships with the aquatic plants and algae that benefit from their activities. By removing suspended particles from the water column, these bivalves increase water clarity and light penetration, enhancing conditions for photosynthesis in submerged vegetation. The mussels benefit from the organic particles they collect, while the clearer water they create supports more diverse and productive plant communities. The reproductive strategies of many freshwater fish create temporary but intensive cooperative relationships that can influence entire ecosystem dynamics. During spawning seasons, multiple species may converge on the same spawning grounds, creating aggregations that provide mutual benefits through shared vigilance against predators and synchronized reproductive timing that overwhelms the capacity of egg and fry predators. Some fish species practice cooperative nest construction, where multiple individuals work together to create and maintain spawning sites that benefit all participants. Bluegill sunfish, for example, often establish colonial nesting areas where the combined efforts of many males create optimal conditions for egg development while providing enhanced protection against nest predators through cooperative defense behaviors. The seasonal migrations of anadromous fish like salmon create moving partnerships that connect freshwater and marine ecosystems across vast geographic ranges. These migrations bring marine-derived nutrients into freshwater systems, supporting not only the fish themselves, but entire communities of terrestrial and aquatic organisms that depend on the annual pulse of ocean-derived energy and nutrients. During their upstream migrations, salmon create disturbance patterns in stream sediments that benefit other species by exposing buried organic matter and creating new habitat features. The carcasses of salmon that die after spawning provide massive inputs of nutrients that support bacterial communities, invertebrate populations, and even terrestrial vegetation along stream corridors. Beaver engineering represents perhaps the most dramatic example of freshwater ecosystem modification through cooperative relationships. While beavers are often viewed as solitary engineers, their dam building activities create habitat conditions that support numerous other species in cooperative relationships. The ponded water behind beaver dams provides habitat for aquatic plants, waterfowl, amphibians, and fish species that cannot survive in fast-flowing streams. The beaver's selective harvesting of woody vegetation creates openings in riparian forests that are quickly colonized by early successional plants, increasing habitat diversity and supporting different communities of insects, birds, and mammals. The organic matter trapped behind beaver dams supports enhanced bacterial and fungal activity, accelerating nutrient cycling and creating productive conditions that benefit entire food webs. Wetland ecosystems created by beaver activity demonstrate complex cooperative relationships between plants adapted to fluctuating water levels and the animal communities they support. Emergent vegetation like cattails and sedges provides nesting habitat for waterfowl, while their root systems help stabilize sediments and improve water quality. The birds, in turn, contribute to seed dispersal and nutrient distribution through their movements between different wetland areas. The symbiotic relationships between freshwater invertebrates and algae reveal sophisticated partnerships that optimize energy, capture, and utilization. Hydras and other cnidarians maintain populations of symbiotic algae within their tissues, gaining photosynthetic products while providing their algal partners with protection and nutrients. These partnerships allow freshwater cnidarians to thrive in nutrient-poor waters where purely predatory lifestyles would be unsustainable. Seasonal dynamics create temporal patterns in freshwater cooperative relationships that reflect the strong seasonal gradients characteristic of temperate and polar aquatic systems. Spring ice breakup and thermal stratification patterns influence the timing of algal blooms, zooplankton population growth, and fish spawning activities, creating windows of opportunity when cooperative relationships can provide maximum benefits. The vertical migration patterns of zooplankton create moving layers of cooperative activity within lake ecosystems. During daylight hours, many zooplankton species retreat to deep, cold waters to avoid visual predators, while at night, they migrate to surface waters to feed on phytoplankton. These migrations create opportunities for cooperative relationships with fish species that time their feeding activities to take advantage of predictable zooplankton movements. Temperature-dependent cooperative relationships in freshwater systems reveal the importance of thermal refugia and microhabitat diversity for maintaining partnership networks. During summer stratification periods, different species may concentrate in specific thermal layers where temperatures and oxygen levels create optimal conditions for their particular cooperative relationships. The role of freshwater cooperative relationships in ecosystem restoration has gained increasing recognition as managers seek to repair damaged aquatic systems. Reintroduction of beaver populations can rapidly restore wetland habitat and the associated cooperative relationships, while restoration of riparian vegetation helps reestablish the terrestrial aquatic connections that support many freshwater partnerships. Urban freshwater systems present unique challenges and opportunities for cooperative relationships. Stormwater management systems that incorporate natural processes can support partnerships between aquatic plants and microbial communities that improve water quality while providing habitat for urban wildlife. Green infrastructure approaches that work with natural cooperative processes often prove more effective and sustainable than purely technological solutions. Climate change affects freshwater cooperative relationships through alterations in precipitation patterns, temperature regimes, and the timing of seasonal events. Changes in snowpack accumulation and melt timing can disrupt the seasonal cues that coordinate cooperative behaviors, while temperature increases may alter the thermal stratification patterns that influence partnership formation in lake ecosystems. The conservation of freshwater cooperative relationships requires watershed-scale approaches that protect the connectivity and natural flow regimes essential for many partnerships. Dam removal projects increasingly recognize that restoring natural flow patterns is essential for reestablishing the cooperative relationships that support healthy aquatic ecosystems. As evening settles over freshwater systems, nocturnal cooperative relationships begin their activities. Aquatic insects emerge from streams and rivers for mating flights that will eventually return nutrients to aquatic systems through egg deposition, while nocturnal fish species form feeding partnerships that take advantage of different sensory capabilities and prey preferences. The gentle sound of flowing water carries the chemical messages and behavioral cues that coordinate freshwater partnerships, reminding us that cooperation flows through aquatic systems as naturally as the water that sustains all life. In the patient work of ecosystem engineers like beavers, in the coordinated activities of spawning fish, in the intimate partnerships between plants and their microbial allies, we find evidence that freshwater cooperation creates the biological richness that makes these systems essential components of Earth's living landscape. In the early morning light filtering through a temperate forest, a mixed flock of songbirds moves through the canopy like a living wave of cooperation. Chickadees lead the procession, their sharp eyes scanning bark crevices for hidden insects, while nuthatches work their way headfirst down tree trunks, exploring territories the chickadees cannot reach. Kinglets hover at branch tips, gleaning tiny arthropods from needle clusters, while creepers spiral up tree trunks in search of prey overlooked by their companions. Each species contributes unique foraging skills to this temporary partnership, creating a collective hunting efficiency that far exceeds what any individual species could achieve alone. Mixed species associations represent one of nature's most dynamic forms of cooperation, where different species band together temporarily to gain advantages that would be impossible through solitary activity. Unlike the permanent partnerships between mycorrhizal fungi and plant roots, or the season long relationships between flowers and their pollinators, these fluid alliances form and dissolve based on immediate circumstances, creating networks of cooperation that adapt constantly to changing opportunities and challenges. The advantages of joining mixed species groups extend far beyond simple strength in numbers. Each participating species brings specialized capabilities that complement those of their temporary partners, creating collective abilities that emerge from diversity itself. In the forest canopy, different bird species possess distinct sensory capabilities, foraging techniques, and habitat preferences that, when combined, provide comprehensive surveillance and resource exploitation across multiple ecological niches simultaneously. The sentinel system that develops within mixed species bird flocks demonstrates how cooperation can enhance survival through shared vigilance. While some flock members focus intensively on foraging, others maintain watchfulness for predators, creating an early warning network that protects all participants. The alarm calls of one species are understood and heeded by others, despite differences in their natural communication systems. This interspecies communication protocol allows the entire flock to benefit from the most acute senses and alertness of its most vigilant members. African savannas showcase some of the most spectacular examples of mixed species cooperation in terrestrial mammals. During the dry season migrations, vast herds of wildebeest, zebras, and gazelles move together across the landscape, their different feeding preferences and habitat requirements, creating a mobile ecosystem that transforms grassland resources more efficiently than any single species could manage. Zebras, with their strong teeth and digestive systems, can process the coarse, tall grasses that other species cannot consume, creating shorter, more nutritious pastures that become accessible to wildebeest and gazelles. The mixed grazing patterns of these African herbivores demonstrate sophisticated resource partitioning that maximizes the carrying capacity of grassland environments. Each species occupies a different stratum of the vegetation community, from the ground level foraging of gazelles to the tall grass consumption of zebras, creating a cascading effect that maintains grassland diversity and productivity. The trampling and grazing patterns of larger species create microhabitat diversity that supports smaller animals and different plant communities. Predator detection advantages multiply dramatically in mixed species herds through the combination of different sensory capabilities and behavioral responses. Zebras possess acute hearing and eyesight that allow them to detect approaching lions or hyenas at considerable distances. Wildebeest excel at detecting scent trails and can identify the recent presence of predators through chemical cues. Gazelles rely on speed and agility, but their nervous, alert behavior serves as an additional early warning system for the entire herd. The temporal dynamics of mixed species associations reveal how cooperation can be precisely timed to maximize benefits while minimizing costs. Many mixed species bird flocks form during autumn and winter when food resources become scarce, and the advantages of cooperative foraging outweigh the increased competition from additional flock members. As spring arrives and territorial breeding behaviors emerge, these cooperative flocks gradually dissolve, demonstrating the flexible nature of these partnerships. Marine environments host their own remarkable examples of mixed species cooperation, particularly in the formation of multi-species schools that provide enhanced protection from predators. Different fish species contribute varying swimming capabilities, sensory acuities, and escape responses that create collective defense mechanisms more effective than any single species could achieve. Fast-swimming species provide rapid escape options, while slower but more maneuverable species offer evasive capabilities that complement the group's overall survival strategy. The phenomenon of nuclear species in mixed species associations demonstrates how certain species can serve as focal points around which cooperative relationships organize. In Amazonian rainforests, army ant swarms attract diverse communities of ant-following birds that feed on insects fleeing from the advancing columns. The ants provide no direct benefit to the birds, but their predictable foraging activities create feeding opportunities that support complex multi-species associations of insectivorous birds. These ant-following assemblages can include dozens of bird species, each exploiting different aspects of the disturbance created by the ant colonies. Some species specialize in catching insects that fly up from the ground. Others focus on prey that attempts to hide in vegetation, and still others glean arthropods from surfaces exposed by the ants' activities. The diversity of foraging strategies within these associations ensures that the feeding opportunities created by ant colonies are utilized comprehensively. The leadership dynamics within mixed species groups reveal sophisticated social organization that transcends species boundaries. In mixed bird flocks, chickadees and titmice often serve as nuclear species that initiate flock movements and attract other species through their contact calls and foraging activities. These core species possess ecological knowledge about resource locations and predator risks that benefits the entire multi-species community. Navigation and route selection in migrating mixed species groups demonstrate how different species can contribute complementary skills to collective decision-making. Some species excel at celestial navigation, others rely on magnetic field detection, and still others use landmark recognition. The combination of these different navigational capabilities creates redundancy and accuracy in route finding that enhances migration success for all participants. The seasonal formation of mixed species feeding aggregations around ephemeral resources showcases how cooperation can maximize exploitation of temporary opportunities. When fruit trees produce synchronized crops, multiple species of frugivorous birds may form temporary associations that allow rapid harvesting before fruits become overripe or are consumed by competitors. These temporary partnerships dissolve once the resource is depleted, only to reform when the next feeding opportunity becomes available. Information transfer within mixed species groups creates knowledge networks that benefit all participants through shared environmental awareness. When one species discovers a new food source or identifies a predator threat, this information spreads rapidly through the group via behavioral cues and vocalizations. Species with superior sensory capabilities or environmental knowledge effectively share these advantages with their temporary partners. The costs and benefits of mixed species association participation create dynamic equilibria that determine group composition and stability. Increased competition for resources must be balanced against enhanced predator protection and foraging efficiency. Species with unique capabilities may find themselves in high demand as group members, while those that contribute less to collective benefits may be excluded from associations during periods of resource scarcity. Climate change affects mixed species associations through alterations in resource distribution, migration timing, and habitat availability. Species that have traditionally formed cooperative associations may find their ranges shifting at different rates, disrupting established patterns of cooperation. Changes in food availability and predator distributions may alter the cost benefit ratios that govern association formation and maintenance. Urban environments create novel opportunities and challenges for mixed species cooperation. City parks and suburban areas may concentrate diverse species in small areas, creating opportunities for associations that would not occur in natural settings. However, urban noise pollution, artificial lighting, and habitat fragmentation can disrupt the communication and coordination mechanisms that enable effective mixed species cooperation. The study of mixed species associations has revealed fundamental principles about cooperation, competition, and community organization that apply broadly across ecological systems. The ability of different species to recognize mutual benefits and coordinate their activities despite differences in communication systems, social structures, and ecological requirements demonstrates the fundamental importance of cooperation in biological communities. Conservation strategies increasingly recognize that protecting mixed species associations requires landscape scale approaches that maintain the habitat connectivity and resource distributions necessary for these dynamic partnerships. Migration corridors, seasonal feeding areas, and roosting sites must be preserved as integrated systems rather than isolated fragments to support the full range of cooperative relationships that depend on them. As dusk settles over landscapes where mixed species cooperation has flourished throughout the day, new associations begin to form. Nocturnal mixed species flocks of insectivorous birds assemble for nighttime foraging, while different combinations of mammalian species emerge to exploit the feeding opportunities that darkness provides. The constant reformation and dissolution of these cooperative relationships demonstrates that diversity itself can be a form of cooperation where differences between species become sources of mutual strength rather than barriers to interaction. In the fluid partnerships that form between unlike species, we discover that cooperation needs no common language, shared ancestry, or identical needs to flourish, requiring only the recognition that working together can achieve what individual effort cannot accomplish alone. In a Costa Rican rainforest, a single strangler fig tree rises above the canopy, its massive crown spreading across an acre of forest. This ancient giant supports a community of extraordinary complexity where hundreds of species have woven their lives into an intricate web of mutual dependence. Epiphytic orchids and bromeliads garden in its branches, creating aerial pools that nurture frogs, insects, and microscopic communities. Birds nest in its hollows and feed on its year-round fruit production, while mammals from tiny bats to large primates depend on its resources. This tree is more than an individual organism. It has become a keystone cooperator whose partnerships structure an entire forest community. The concept of cooperative communities represents ecology's recognition that partnerships between species can become so fundamental to ecosystem function that they define the very structure and stability of biological systems. These are not simply collections of individual cooperative relationships existing side by side, but integrated networks where the success of each partnership depends on the success of many others, creating emergent properties that exist only at the community level. Keystone mutualists like the strangler fig demonstrate how certain cooperative relationships become so essential that they support entire webs of secondary partnerships. The fig's relationship with its specialized pollinating wasps ensures continuous fruit production throughout the year, providing a reliable food source during the seasonal fruit scarcity that characterizes many tropical forests. This dependable resource base enables the fig to support populations of frugivorous animals that might otherwise be unable to persist through lean periods. The architecture of cooperation within these communities reveals multiple layers of interdependence that create remarkable stability and resilience. The fig's massive canopy provides structural support for epiphytic communities that would otherwise be unable to survive in the understory's low-light environment. The epiphytes, in turn, create habitat complexity that supports diverse arthropod communities, which provide food for insectivorous birds and small mammals that contribute to seed dispersal and forest regeneration processes. Water collection and cycling within these aerial communities demonstrates how cooperative relationships can create their own supporting infrastructure. Bromeliad plants growing as epiphytes collect rainwater in their leaf bases, creating thousands of small aquatic habitats throughout the forest canopy. These aerial ponds support communities of mosquito larvae, tree frogs, and microscopic organisms that would otherwise be restricted to terrestrial water sources. The nutrient cycling processes within cooperative communities reveal sophisticated mechanisms for resource conservation and sharing that maximize the efficiency of ecosystem function. Epiphytic roots and specialized bacterial communities intercept nutrients from canopy throughfall and animal waste, preventing their loss to the forest floor and making them available to the broader aerial community. This canopy level nutrient retention system supports productivity levels that would be impossible if each species relied solely on resources obtained from soil sources. Coral reef ecosystems exemplify cooperative communities where the foundational partnership between corals and their algal symbionts creates the physical and biological infrastructure that supports extraordinary biodiversity. The three-dimensional structure created by coral skeletons provides habitat complexity that enables countless other cooperative relationships to develop, from cleaning stations to territorial partnerships between different fish species. The calcium carbonate matrix of coral reefs creates a shared resource that benefits the entire community while being maintained through the collective activities of many species. Boring organisms create cavities and crevices that provide shelter for numerous species, while their waste products contribute nutrients that support the reef's overall productivity. Corallivorous fish that feed on coral polyps may seem destructive, but their grazing activities prevent competitive algal overgrowth and maintain the diversity of coral species within reef communities. Nutrient cycling within coral reef communities involves complex partnerships between species at multiple trophic levels. Cleaner fish that remove parasites from larger fish transport nutrients between different parts of the reef system through their movements and waste products. Seabirds that nest on islands adjacent to reefs contribute marine-derived nutrients that support terrestrial vegetation, which in turn provides shade and organic matter that influences nearshore reef conditions. Prairie ecosystems demonstrate how cooperative communities can be dominated by underground partnerships that create emergent properties invisible from surface observations. The extensive mycorrhizal networks connecting grass species create shared resource pools that enable prairie communities to withstand droughts, fires, and grazing pressures that would destroy systems dominated by individual species competing independently. The fire-adapted nature of prairie cooperative communities reflects millions of years of coevolution between plant partnerships and natural disturbance regimes. The underground storage organs and extensive root systems, maintained through mycorrhizal partnerships, allow prairie plants to resprout rapidly after fires, while the rapid post-fire growth creates conditions that favor the reestablishment of fungal networks and associated cooperative relationships. Grazing relationships within prairie ecosystems create additional layers of cooperation that influence community structure and function. Large herbivores like bison create patchy grazing patterns that maintain habitat diversity and prevent competitive exclusion, while their trampling and wallowing activities create microhabitats that support specialized plant and animal communities. The nutrient inputs from herbivore waste are distributed through mycorrhizal networks, creating hotspots of productivity that benefit entire plant communities. Forest cooperative communities reveal how partnerships between trees and their associated organisms create ecosystem properties that persist across centuries or millennia. Old-growth forests develop cooperative networks of extraordinary complexity where individual trees become integrated into community-level systems for resource sharing, communication, and mutual support. The death of individual trees creates opportunities for new partnerships to develop while contributing nutrients and habitat structure that benefit the broader community. The understory communities of mature forests depend on cooperative relationships with canopy species that modify light, temperature, and moisture conditions to create suitable habitat for shade-adapted species. Mycorrhizal networks connect understory plants to canopy trees, enabling the sharing of resources that allows diverse plant communities to coexist under low-light conditions, where photosynthetic productivity would otherwise be insufficient to support such diversity. Succession in forest communities involves the sequential development of different cooperative networks as environmental conditions change and new partnership opportunities emerge. Pioneer species establish initial mycorrhizal relationships and begin soil development processes that create conditions suitable for more specialized cooperative relationships. As forests mature, increasing habitat complexity enables more diverse and sophisticated partnerships to develop. Wetland cooperative communities demonstrate how partnerships between terrestrial and aquatic species can create highly productive ecosystems that provide essential services to broader landscapes. The relationships between emergent vegetation, waterfowl, and microbial communities create nutrient cycling processes that maintain water quality while supporting diverse biological communities. Beaver engineering activities create habitat mosaics that support different cooperative networks adapted to varying water depths and hydro periods. The seasonal dynamics of wetland cooperative communities reflect the adaptability of partnership networks to changing environmental conditions. During high water periods, aquatic partnerships dominate, while low water periods favor terrestrial cooperative relationships. Many species maintain partnerships that function across these transitions, creating continuity that stabilizes community structure despite fluctuating conditions. Pollinator networks within plant communities create cooperative webs that extend across landscapes and connect different habitat types. The flowering sequences of different plant species ensure continuous nectar and pollen availability throughout growing seasons, while pollinator movements create genetic connections between plant populations that maintain species diversity and adaptive potential. The decline of pollinator populations demonstrates how the breakdown of keystone cooperative relationships can cascade through entire communities, affecting not only the immediate partners involved, but also the numerous secondary relationships that depend on them. Plant communities that lose their primary pollinators may experience reduced reproduction and genetic diversity, which in turn affects the herbivores, seed dispersers, and other organisms that depend on these plants. Conservation of cooperative communities requires landscape scale approaches that maintain the connectivity and environmental conditions necessary for partnership networks to function across spatial and temporal scales. Protected areas must be large enough to encompass the full range of cooperative relationships within communities, while corridors between protected areas enable the movement of organisms and genetic material necessary to maintain partnership networks. Restoration of degraded cooperative communities focuses on reestablishing the foundational partnerships that structure community development. Reintroduction of keystone mutualists, restoration of mycorrhizal networks, and reestablishment of natural disturbance regimes can initiate the recovery of cooperative relationships that support broader community restoration. Climate change challenges cooperative communities through its effects on the environmental conditions that maintain partnership networks and the synchronization of cooperative activities. Changes in temperature and precipitation patterns may disrupt the timing of cooperative interactions, while shifts in species ranges may separate partners that have co-evolved over long time periods. Urban environments create opportunities for novel cooperative communities to develop as species adapt to human-modified landscapes. Urban forest fragments, constructed wetlands, and green infrastructure can support cooperative networks that provide ecosystem services while maintaining biodiversity in developed areas. As night settles over the world's cooperative communities, the networks of partnership that structure these systems continue their ancient rhythms. In coral reefs and rainforest canopies, in prairie grasslands and wetland margins, the web of cooperative relationships that creates biological community continues its patient work of mutual support and shared benefit. These communities remind us that cooperation is not merely a strategy used by individual species, but a fundamental organizing principle that creates the very fabric of biological community and ecological stability. As the first warm days of spring arrive in a temperate woodland, an invisible transformation begins beneath the forest floor. Mycorrhizal networks that lay dormant through winter months suddenly pulse with renewed activity as tree roots begin their annual growth surge. Chemical signals flow through fungal pathways, coordinating the emergence of wildflowers with the availability of nutrients released from decomposing autumn leaves. Above ground, migrating birds return to time their arrival with the peak emergence of insects, while flowering plants prepare to synchronize their blooms with the activity patterns of their pollinating partners. This is the great awakening of seasonal cooperation, where millions of partnerships must coordinate their activities with the rhythms of Earth's changing seasons. The seasonal timing of cooperative relationships represents one of nature's most remarkable feats of coordination, requiring partners to synchronize their activities across vastly different life cycles and biological rhythms. Unlike the constant partnerships of tropical ecosystems, temperate and polar regions demand flexible, cooperative strategies that can adapt to dramatic seasonal changes in temperature, daylight, and resource availability. Spring emergence patterns reveal the sophisticated timing mechanisms that govern seasonal partnerships. As soil temperatures rise and daylight hours lengthen, dormant mycorrhizal fungi begin producing the enzymes and transport proteins necessary to resume nutrient exchange with their plant partners. The timing of this reactivation must precisely match the beginning of root growth in trees and the germination of herbaceous plants, requiring coordination mechanisms that have been refined over millions of years of coevolution. The sequence of spring wildflower blooms demonstrates temporal resource partitioning that maximizes pollinator services while minimizing competition between plant species. Skunk cabbage and bloodroot bloom in the cold soils of early spring when few competitors are active, but before deciduous trees leaf out and reduce understory light levels. Trout lilies and spring beauties follow as temperatures warm, while later species like wild ginger and mayapple time their flowering to coincide with peak activity of their specialized pollinators. Pollinator emergence patterns show remarkable synchronization with their plant partners' flowering schedules. Specialist bees emerge from overwintering sites at precisely the time when their preferred flowers become available, often with timing accuracy measured in days rather than weeks. This synchronization requires both partners to respond to the same environmental cues, creating natural coordination mechanisms that maintain partnership effectiveness despite year-to-year variation in seasonal timing. The seasonal migration of pollinators creates moving networks of cooperation that connect plant communities across vast geographic ranges. Hummingbirds traveling north from Central American wintering grounds follow corridors of flowering plants that provide refueling opportunities precisely when the birds need them most. The timing of flowering along these migration routes has co-evolved with hummingbird migration schedules, creating continental-scale cooperation networks that span multiple countries and ecosystems. Summer partnerships reflect the peak resource availability that characterizes the growing season in temperate regions. Mycorrhizal networks reach maximum activity levels as both plant and fungal partners invest heavily in growth and reproduction. The high rates of photosynthesis in summer active plants provide abundant carbohydrates for fungal partners, while the extensive hyphal networks developed during this season maximize nutrient capture for plant growth and seed production. The complexity of summer cooperative relationships increases as habitat diversity reaches its annual maximum. Temporary partnerships form around ephemeral resources like fruiting trees or insect emergence events, while longer-term relationships, like those between plants and their mycorrhizal partners, operate at peak efficiency. The layered canopy structure of summer forests creates multiple cooperation zones, from ground-level partnerships between herbs and soil organisms to canopy relationships between epiphytes and their host trees. Autumn preparations for winter reveal the forward-looking nature of many cooperative relationships. Trees begin transferring resources to their mycorrhizal partners, providing fungi with the carbon stores they will need to survive winter months when photosynthesis ceases. Seed dispersal partnerships intensify as plants race to complete reproduction before killing frosts, while animals that depend on these partnerships work to gather and cache the resources they will need during winter months. The timing of leaf senescence and abscission creates pulses of organic matter that fuel decomposer communities and soil based cooperative relationships throughout winter. The chemical composition of different leaf species influences the types of decomposer organisms that colonize them, creating temporal niches that support diverse microbial communities during the resource scarce winter period. Winter partnerships represent cooperation under the most challenging conditions, where survival often depends on maintaining beneficial relationships despite severely limited resources. Evergreen trees continue photosynthesis at reduced rates, providing minimal but essential carbon inputs to their mycorrhizal partners. These winter partnerships may operate at barely detectable levels, but they provide the foundation for rapid reactivation when spring conditions return. The formation of winter aggregations demonstrates how seasonal stress can intensify cooperative relationships. Mixed species bird flocks become more cohesive and long lasting during winter months, when the benefits of cooperative foraging and predator detection outweigh the costs of increased competition for limited food resources. These winter partnerships often dissolve as spring arrives and territorial breeding behaviors reassert themselves. Seed dormancy and germination timing reflect cooperative relationships between plants and their seasonal environment. Many seeds require specific sequences of temperature and moisture conditions that ensure germination occurs when mycorrhizal partners are active and ready to form beneficial associations. The timing mechanisms that control seed dormancy have co-evolved with seasonal patterns of fungal activity, creating coordination between partners that may be separated by months or even years. The seasonal dynamics of aquatic cooperative relationships reflect the strong thermal stratification patterns that characterize temperate lakes and rivers. Spring turnover events redistribute nutrients and oxygen throughout water columns, reactivating partnerships between algae and bacteria that lay dormant during winter months. Summer stratification creates distinct zones where different cooperative relationships operate, while autumn turnover redistributes the products of summer partnerships throughout aquatic systems. Ice formation and melting create dramatic, seasonal transitions that require flexible partnership strategies. Aquatic plants that maintain winter partnerships with ice-dwelling bacterial communities must rapidly shift to open water relationships as ice melts and growing conditions change. The timing of these transitions influences the success of many aquatic partnerships and determines the seasonal productivity of entire freshwater ecosystems. Migration partnerships demonstrate cooperation across continental scales, where seasonal movements connect breeding and wintering areas through networks of stopover sites that provide essential resources for traveling animals. The timing of resource availability at these stopover locations must be precisely coordinated with migration schedules, creating intercontinental cooperation networks that depend on seasonal coordination across multiple time zones and climatic regions. Climate change poses unprecedented challenges to seasonal cooperation by disrupting the environmental cues that coordinate partnership timing. Rising temperatures advance the onset of spring in many regions, but different species respond to different cues, creating mismatches between partners that have co-evolved for millions of years. Flowering plants that respond primarily to temperature may bloom before their pollinators, which respond to day length, emerge from winter dormancy. The phenological shifts caused by climate change create ripple effects throughout seasonal cooperation networks. Early spring flowering may provide inadequate pollination services if pollinator emergence remains tied to day length rather than temperature. Late arriving migrants may find that peak food resources have already passed, disrupting partnerships that connect breeding success to seasonal resource availability. Adaptation to changing seasonal patterns requires flexibility in partnership timing that may take many generations to develop. Species with short generation times may adapt relatively quickly to shifting seasonal conditions, while long lived species with complex life cycles may struggle to adjust their partnership timing rapidly enough to keep pace with environmental change. Conservation strategies for seasonal cooperative relationships must account for the temporal complexity of these partnerships and the environmental cues that coordinate their timing. Protecting seasonal cooperation requires maintaining not only the habitats where partnerships occur, but also the environmental conditions that provide timing, cues, and the connectivity that enables seasonal movements and resource access. Restoration of seasonal cooperative relationships focuses on reestablishing the environmental conditions and timing mechanisms that coordinate partnership activities. This may involve restoring natural hydrology that provides seasonal flooding cues, maintaining temperature regimes that coordinate emergence timing, or preserving migration corridors that connect seasonal partnerships across landscapes. As winter settles over temperate landscapes, the annual cycle of seasonal cooperation prepares for another round of coordination and partnership. Dormant mycorrhizal networks maintain minimal activity that will enable rapid spring reactivation, while overwintering animals prepare for the partnerships that will sustain them through the coming year. Seeds lie buried in soil, their germination timing precisely calibrated to coordinate with the seasonal availability of their microbial partners. The quiet rhythms of seasonal preparation remind us that cooperation in nature requires not only finding the right partners, but timing partnerships to match the ever changing opportunities and challenges that define life in seasonal environments. In the patient synchronization of seasonal cooperative relationships, we find evidence that nature's partnerships are not fixed arrangements, but dynamic relationships that flow and adapt with the changing rhythms of our turning planet. In the aftermath of a forest fire that swept through Montana's wilderness, blackened tree trunks stand like monuments to destruction across a landscape seemingly devoid of life. Yet beneath the charred surface, an invisible network of recovery is already beginning. Mycorrhizal fungal spores that survive the flames in deep soil layers start to germinate, seeking out the first green shoots of fire-adapted plants. These underground partnerships will form the foundation of forest recovery, demonstrating that nature's healing processes are fundamentally cooperative endeavors where restoration emerges not from individual effort, but from the patient rebuilding of relationships between species. The science of ecological restoration has gradually recognized that damaged ecosystems heal most effectively when restoration efforts focus on reestablishing the cooperative relationships that structure healthy communities rather than first simply replanting individual species. This partnership based approach to healing acknowledges that ecosystems are not merely collections of species occupying shared space, but integrated networks of cooperative relationships where the success of restoration depends on rebuilding the invisible connections that allow communities to function as unified wholes. Fire-adapted ecosystems demonstrate nature's own restoration strategies, where cooperative relationships have evolved to not only survive disturbance, but to use it as an opportunity for renewal and regeneration. In the first growing season after a fire, nitrogen-fixing plants often dominate early succession communities, their bacterial partners working to rebuild soil nitrogen levels depleted by the intense heat. These pioneer cooperators create the chemical foundation that will eventually support more complex plant communities and their associated partnerships. The role of mycorrhizal networks in forest restoration reveals how underground cooperation can drive above ground recovery. Surviving fungal networks that persisted in protected microsites or as dormant spores begin extending their hyphal connections to newly germinating plants, providing essential nutrients and water that enable rapid establishment in the harsh post-fire environment. These fungal partners effectively serve as biological bridges between the old forest community and the new one developing in its place. Seed dispersal partnerships become crucial during early restoration phases as surviving animals transport seeds from undamaged areas into recovering zones. Birds that nested in forest edges or survived the fire in refugia begin carrying seeds of forest plants back into burned areas, while mammals create scatter-hoarding caches that establish new patches of vegetation. These mobile partnerships ensure that genetic material from healthy populations reaches restoration sites where it can contribute to community recovery. Wetland restoration projects have discovered that success depends heavily on reestablishing the partnerships between aquatic plants and their associated microbial communities. Simply replanting emergent vegetation without considering the soil bacteria and mycorrhizal fungi that support plant health often results in poor establishment and slow recovery. Restoration efforts that inoculate restoration sites with beneficial microorganisms from healthy wetlands achieve much higher success rates and faster ecosystem development. The restoration of pollination networks requires coordinated efforts that address both plant and pollinator needs simultaneously. Establishing native wildflower meadows provides only temporary benefits if the soil lacks the microbial communities that support plant health, or if the surrounding landscape cannot support stable pollinator populations. Successful restoration creates habitat corridors and resource patches that maintain pollinator populations while providing the continuous flowering sequences that sustain these populations throughout growing seasons. Prairie restoration exemplifies partnership-based healing on a landscape scale, where the reestablishment of mycorrhizal networks becomes the foundation for rebuilding entire grassland communities. Traditional restoration approaches that focused on seed mixes and fire management often produced plant communities that looked correct but lacked the underground cooperation networks that characterize healthy prairies. Modern restoration incorporates soil transfers from healthy prairies that contain the full spectrum of beneficial microorganisms needed to support native plant partnerships. The timing of restoration activities reflects an understanding of seasonal cooperation patterns and the need to coordinate restoration interventions with natural partnership rhythms. Seeding activities timed to coincide with peak mycorrhizal activity increase establishment success, while pollinator habitat restoration scheduled to provide resources during critical life cycle phases ensures that restoration efforts support stable rather than temporary population recovery. Coral reef restoration has evolved from efforts to transplant individual coral colonies to comprehensive approaches that rebuild the cooperative relationships between corals and their algal symbionts. Temperature stress and ocean acidification often disrupt the fundamental partnerships that enable coral growth, so restoration efforts now focus on identifying and propagating coral algae combinations that can maintain their partnerships under changing environmental conditions. The restoration of cleaning station networks within coral reef systems demonstrates the importance of cooperative relationships in ecosystem recovery. Simply transplanting corals without considering the fish and invertebrate communities that maintain reef health through cleaning services results in communities vulnerable to disease and parasitism. Successful restoration creates habitat complexity that supports cleaner organisms while providing the territorial structure necessary for cleaning stations to function effectively. Urban ecosystem restoration presents unique challenges that require novel approaches to partnership building in human modified environments. Green infrastructure projects that support mycorrhizal networks, pollinator corridors, and urban forest partnerships can create cooperative communities that provide ecosystem services while supporting biodiversity in developed areas. These urban partnerships often involve species combinations that would not occur in natural settings, but provide similar ecological functions under urban conditions. The restoration of riparian ecosystems along degraded waterways demonstrates how partnership based approaches can address multiple environmental challenges simultaneously. Reestablishing vegetation alone provides limited benefits if soil microbial communities remain degraded, but restoration that includes beneficial bacteria and mycorrhizal fungi creates plant communities capable of stabilizing banks, filtering pollutants, and supporting aquatic organisms through improved water quality. Climate change adaptation requires restoration approaches that build resilient partnerships capable of functioning under changing environmental conditions. Traditional restoration that aims to recreate historical communities may fail if those communities' cooperative relationships cannot function under altered temperature and precipitation regimes. Forward looking restoration identifies partnership combinations that can maintain ecosystem functions while adapting to environmental changes. Assisted migration programs increasingly recognize that moving species to new locations requires establishing the cooperative relationships that enable them to thrive in novel environments. Relocating plants without their mycorrhizal partners often results in poor establishment, while moving animals without considering their food sources and habitat requirements rarely achieves conservation goals. Successful assisted migration transfers entire cooperation networks rather than individual species. The role of citizen science in partnership based restoration creates opportunities for public engagement that goes beyond traditional volunteer planting activities. Community members can participate in monitoring mycorrhizal network health, documenting pollinator plant relationships, and maintaining seed banks that preserve the genetic diversity necessary for successful partnership restoration. These activities create connections between human communities and the natural partnerships they are working to restore. Monitoring and evaluation of restoration success increasingly focuses on the development of cooperative relationships rather than simple measures of plant survival or animal abundance. Healthy partnerships between plants and their microbial communities produce measurable changes in soil chemistry and structure, while successful pollination networks result in seed production and genetic diversity that can be tracked over time. These partnership indicators provide early warnings of restoration problems and guide adaptive management strategies. The economic benefits of partnership based restoration often exceed the costs when ecosystem services are considered comprehensively. Restored mycorrhizal networks improve soil stability and reduce erosion, while re-established pollination networks support both wild plant communities and agricultural systems. Wetland partnerships provide water filtration services, while urban forest partnerships reduce energy costs and improve air quality. These economic arguments help justify the additional investments required for partnership based restoration approaches. Research into restoration partnerships continues to reveal new opportunities for cooperative approaches to ecosystem healing. Understanding how different microbial communities support plant establishment under various conditions enables more precise restoration strategies, while knowledge of pollinator movement patterns informs landscape scale habitat design. Advances in molecular techniques allow restoration practitioners to monitor partnership development and adjust management strategies based on the success of cooperative relationship establishment. The psychological benefits of participating in partnership based restoration create positive feedback loops that sustain long term restoration efforts. Community members who understand the cooperative relationships they are helping to restore often develop deeper connections to restoration sites and greater commitment to long term stewardship. Educational programs that highlight partnership restoration create environmental advocates who understand ecosystem function at a fundamental level. As restoration sites mature and cooperative relationships establish themselves, the process of healing becomes self sustaining. Mycorrhizal networks expand and support new plant establishment, while pollination networks create the genetic diversity necessary for adaptation to local conditions. Seed dispersal partnerships spread restoration benefits beyond original project boundaries, creating expanding zones of cooperative recovery that demonstrate nature's capacity for self renewal when given appropriate support. The gentle work of partnership restoration reminds us that healing damaged ecosystems requires patience, understanding, and respect for the cooperative relationships that make ecological communities possible. In the slow rebuilding of trust between species, in the careful reconstruction of communication networks, in the patient restoration of mutual benefit, we find models for healing that extend far beyond ecological applications to encompass all relationships that sustain life's abundance and resilience. In the deepening twilight of a summer evening, as the last light fades from a meadow where countless partnerships have played out their ancient rhythms throughout the day, the interconnected nature of all cooperative relationships becomes luminously clear. The mycorrhizal networks that connected grassroots to soil fungi continue their quiet nutrient exchanges in the darkness. Night-flying moths begin their visits to evening-blooming flowers, continuing pollination services that their day-shift butterfly counterparts have temporarily suspended. In streams that border the meadow, cleaning partnerships between fish continue under starlight, while in the forest beyond, mixed-species flocks of nocturnal birds form temporary alliances for nighttime foraging. This seamless transition from day to night partnerships reveals a profound truth. Cooperation is not an occasional strategy employed by individual species, but the fundamental organizing principle that creates and sustains the harmony of life itself. The networks of cooperation that we have explored throughout this journey demonstrate that partnership and mutual aid are not exceptions to nature's rules, but expressions of life's deepest organizing principles. From the molecular partnerships that enable nitrogen fixation in soil bacteria to the landscape scale cooperation networks that connect entire continents through migration corridors, We find that living systems achieve their greatest stability, productivity, and resilience through collaborative relationships rather than competitive struggle. The prevalence of cooperative relationships across all scales of biological organization suggests that collaboration may be evolution's most successful strategy for dealing with the fundamental challenges of life. Individual organisms face constant pressures from resource limitation, environmental uncertainty, and the need to reproduce successfully in competitive environments. The countless partnerships we have examined represent evolution's creative solutions to these challenges, demonstrating repeatedly that working together produces outcomes that individual effort cannot achieve. The chemistry of cooperation reveals itself as a universal language that transcends the boundaries between species, kingdoms, and even the traditional divisions between living and non-living systems. The molecular signals that coordinate mycorrhizal partnerships operate through the same basic principles as the chemical communication that enables cleaner fish to advertise their services or flowers to attract pollinators. This biochemical foundation of cooperation suggests that partnership may be as fundamental to life as metabolism or reproduction. The architecture of cooperative networks demonstrates emergent properties that exist only at the community and ecosystem levels, where the interactions between multiple partnerships create capabilities that no individual relationship could generate alone. Coral reefs exemplify this emergence, where the foundational partnership between corals and algae creates the physical structure that enables thousands of other cooperative relationships to develop, resulting in biological communities of extraordinary complexity and productivity. The resilience of cooperative systems provides compelling evidence that partnership-based organization creates stability through redundancy and mutual support rather than through competitive dominance. When disturbances disrupt individual partnerships, the network effects of cooperative communities often enable rapid recovery through the support of unaffected relationships. Forest ecosystems recovering from fire demonstrate this resilience, where surviving mycorrhizal networks help reestablish plant communities that then support the recovery of pollination networks, seed dispersal partnerships, and ultimately the full spectrum of cooperative relationships that characterize healthy forests. The evolutionary persistence of cooperative relationships across hundreds of millions of years reveals their fundamental importance to life's success on Earth. The partnerships between fungi and plant roots that we see in modern forests are variations on themes that first appeared when plants colonized terrestrial environments over 400 million years ago. The cleaning relationships between fish species reflect behavioral patterns that have been refined through countless generations of natural selection, demonstrating that cooperation provides genuine evolutionary advantages that persist across vast spans of geological time. The global reach of cooperative networks creates connections that span continents and ocean basins, linking local partnerships into planetary scale systems that regulate Earth's climate, chemical cycles, and biological productivity. The migration partnerships that connect Arctic breeding grounds to tropical wintering areas demonstrate how local cooperative relationships contribute to global patterns of biological organization. These continental scale networks remind us that even the most intimate partnerships between individual organisms contribute to the functioning of Earth's biosphere as an integrated system. The seasonal rhythms of cooperation reflect life's adaptation to the astronomical cycles that govern Earth's climate, showing how partnerships must synchronize not only with each other, but with the fundamental rhythms of planetary motion. The precise timing that coordinates spring emergence of mycorrhizal activity with plant growth, or the synchronization of flowering with pollinator emergence, demonstrates that cooperation extends beyond relationships between organisms to encompass relationships with the physical environment itself. The healing power of cooperative relationships provides hope for addressing the environmental challenges that face our planet. Understanding that ecosystem recovery depends on reestablishing partnership networks rather than simply protecting individual species offers new approaches to conservation and restoration that work with natural processes rather than against them. The success of partnership based restoration projects demonstrates that cooperative principles can guide human efforts to heal damaged ecosystems and create sustainable relationships with the natural world. The beauty of cooperative relationships reminds us that the aesthetic qualities we find most appealing in nature often reflect the visible expressions of underlying partnerships. The colors and patterns of flowers evolved as advertisements to attract pollinating partners, while the intricate structures of coral reefs represent the accumulated efforts of countless generations of coral algae partnerships. The scenes that move us most deeply in nature are often displays of cooperation in action, suggesting that our aesthetic sense may be attuned to recognizing the harmony that partnership creates. The efficiency of cooperative systems challenges assumptions about the necessity of competition as a driving force in natural selection. While competitive interactions certainly exist in nature, the partnerships we have explored demonstrate that cooperation can achieve resource acquisition, reproduction, and survival more effectively than individual effort. The energy efficiency of mycorrhizal partnerships, the reproductive success of plants with effective pollinators, and the survival advantages of mixed species animal groups all provide evidence that cooperation often outperforms competition as a survival strategy. The universality of cooperative relationships across different types of environments, from the deepest ocean trenches to the highest mountain peaks, suggests that partnership represents a fundamental property of life itself rather than an adaptation to specific environmental conditions. Whether in the extreme conditions around hydrothermal vents or the challenging environment of Arctic tundra, life finds ways to create cooperative relationships that enable survival and reproduction under conditions that would be impossible for individual organisms to endure alone. The temporal scales of cooperative relationships span from brief encounters lasting minutes to partnerships that persist for centuries or millennia, demonstrating that cooperation operates across all relevant timescales in biological systems. The cleaning interactions between fish last only moments, while the mycorrhizal networks in old-growth forests represent partnerships that have been maintained continuously for thousands of years. This temporal diversity shows that cooperation can be both immediately beneficial and a long-term investment in stable, productive relationships. The complexity that emerges from simple cooperative interactions reveals cooperation as a creative force that generates biological diversity and ecological complexity. The basic principle of mutual benefit, expressed through countless variations in different environmental contexts, has produced the extraordinary diversity of partnerships that characterize Earth's ecosystems. From the molecular partnerships within lichen bodies to the landscape-scale networks that support migrating animals, cooperation has been evolution's primary tool for creating biological complexity. The future of cooperative relationships depends on our recognition that human activities increasingly influence the environmental conditions that make natural partnerships possible. Climate change, habitat fragmentation, and pollution all pose threats to the delicate timing, chemical communication, and environmental stability that many partnerships require. Understanding and protecting cooperative relationships represents one of our most important responsibilities as members of Earth's biological community. As we conclude this exploration of nature's partnerships, we find ourselves surrounded by evidence that cooperation is not merely one strategy among many that life employs, but the fundamental process through which life creates order, beauty, and abundance from the basic materials of Earth's biosphere. In every breath we take, oxygen produced through the ancient partnership between cyanobacteria and their environment sustains our lives. The food we eat exists because of partnerships between plants and their pollinators, their mycorrhizal fungi, their nitrogen-fixing bacterial allies. The stability of Earth's climate depends on cooperative relationships between organisms and geochemical processes that have regulated atmospheric composition for billions of years. In recognizing the centrality of cooperation to life's success, we discover not only how nature works, but perhaps how we ourselves might learn to work with nature and with each other to create a future characterized by harmony rather than conflict, by mutual support rather than competitive struggle, by the patient building of relationships rather than the destructive pursuit of individual advantage. The gentle wisdom of partnership that flows through every ecosystem on Earth offers a model for human cooperation that could transform our relationship with the natural world and with each other. In the quiet partnerships that continue their work as we sleep, in the seasonal rhythms of cooperation that will continue long after we are gone, we find evidence of life's deepest truth. That we exist not as separate individuals struggling against the world, but as participants in an ancient network of mutual support that connects every living thing in relationships of profound interdependence and enduring cooperation.